Okay, let's go ahead and get started. Good morning. I'm Bert Semler, Director of the UCI Center for Virus Research. On behalf of Sanghyuk Shin, Director of the UCI Infectious Disease Science Initiative, I welcome you to our jointly sponsored web symposium, COVID-19 Dynamics, State of the Science. Given the local, national, and international devastation caused by the COVID-19 pandemic, we are all acutely aware of the need to better understand this very complex disease and the properties of the novel coronavirus that underlie the disease symptoms and dynamics. Today, we'll have 6 presentations from research experts covering a range of key COVID-19 topics, including viral pathogenesis, animal models of disease, therapeutics and vaccines, immune response, virus transmission dynamics, epidemiology, and viral evolution and genomics. The fundamental knowledge gained from the different areas of research described by today's speakers will be critical to our understanding of this newly emerged, highly transmissible coronavirus and how it has led to such high levels of morbidity and mortality. The knowledge will also inform our ability to design antiviral therapeutics and vaccines that are desperately needed to control the SARS-CoV-2 infections. So before we get started with the scientific presentations, I'd like to acknowledge Michael Bookmeyer, Ilhem Messaoudi, and Guiyan Yang for their important help in putting the scientific program together. I'd also like to acknowledge the crucial logistics support provided by Alyssa Sanchez from the University Advancement, Crystal Spitali from the Center for Virus Research, JP Clark and Alice Manning from the Office of the Provost, Will Alvarez from UCI Media, and Megan Barissima and Xiao Wang from Infectious Disease Science Initiative. So for today's program, we will have audio for all attendees muted to eliminate any background noise or signal interference. For the Q&A session that follows each presentation, please use the Q&A function on your Zoom screen. And FYI, this symposium will be recorded. So now, I'd like to introduce the moderator of our first session, Matthew Marsden, a newly arrived assistant professor in the Department of Microbiology and Molecular Genetics and Division of Infectious Diseases here at UCI. Matt?
Thank you, Dr. Semler. So it's my pleasure to introduce the first speaker of the day, who is Dr. Stanley Perlman from the University of Iowa, who's a professor of microbiology and immunology and also pediatrics. Dr. Perlman received his PhD in biophysics from MIT in Massachusetts and his MD from the University of Miami in Florida. He was trained in pediatrics and pediatric infectious diseases at Boston Children's Hospital in Massachusetts. So his current research efforts are focused on coronavirus pathogenesis, including virus-induced demyelination and SARS, MERS, and COVID-19 diseases. So Dr. Perlman's laboratory has developed several novel animal models useful for studying pathogenesis and evaluating vaccines and antiviral therapies. His studies are directed at understanding why aged patients and mice develop more severe disease than younger individuals after infection with these viruses, and also why there's a male predominance in patients with more severe disease after infection. He and his colleagues demonstrated the transduction of mice with an adenovirus expressing the human receptor for MERS-CoV, DPP4, rendered them sensitive to infection, providing the first rodent model useful for studying MERS. Similar approaches have also been used to develop a small mouse model for COVID-19. He's also developed models for the loss of sense of smell observed in patients with COVID-19. So Dr. Perlman's previous studies have proven particularly important in guiding our understanding of the current COVID-19 pandemic, as evidenced by the nearly 10,000 citations his prior work has received this year alone. So with that, I would like to welcome Dr. Perlman, who will present on the topic of coronavirus pathogenesis.
Thank you, Matt. So let me share my screen here. Yes, so what I think I would like to start by emphasizing what Matt said, which is that those of us who have been studying coronaviruses for many, many years, and which of course includes Mike Buckmeyer on the Irvine faculty at Tom Lane, know that this epidemic has many of the features that one might have learned from prior both human and non-human coronavirus infections. And I'll try to point out some of those as we go along, but I won't have time to point out all of them. So coronaviruses, just to very briefly introduce the virus, the large RNA viruses There's a broad diversity across mammalian and avian species. They're very large viruses at the size of 31 kb, yet on the surface, they seem to do the same, have the same amount of genetic information or same amount of genetic potential as a virus like poliovirus or HIV, which are a quarter to a third as big as a coronavirus. So the question is, why are these viruses so big? This is something we still don't really understand. Maybe this helps at crossing species, which these viruses do very well. Before SARS and MERS and COVID-19 virus came about, we really only knew of two viruses, OC43 and 229E. They caused the common cold. And then after SARS was recognized, other investigators found NL63 and HKU1, which are also causes of the common cold. And of course, we know about SARS and MERS, which are recent zoonoses with pandemic potential. And the SARS one has been realized, not as SARS per se, but by its relative SARS-CoV-2. And MERS, of course, is also found in bats in various forms. MERS itself is not likely to become pandemic because it's really a camel virus and it crosses from camels to people. But the concern is that there's going to be a MERS-2 lurking somewhere that will have pandemic potential. And I already mentioned that it's a large virus. The kinds of diseases that this caused before we had the severe respiratory diseases in humans was really noted for causing diarrheal diseases in pigs and in the lab and in other animals, cows, for example. And pigs seem to be particularly susceptible to various coronaviruses because for many years, transmissible gastroenteritis virus was a disease of pigs that caused major losses in suckling pigs who developed diarrhea. That was eliminated by actually a vaccine developed by nature. And then after that, we've had porcine epidemic diarrhea virus and porcine Delta coronavirus, and a new one in China now, a severe acute diarrhea syndrome virus. And these are all coronaviruses. And the last one, SADS, is really interesting because it seems to be it resulted from a direct infection of bats onto pigs. So I'm not going to have time to talk about that much, but it's another example of these viruses crossing from bats to infect a different species. And in the laboratory, for many years, the main studies of coronaviruses were a mouse hepatitis virus because they're causes of hepatitis and demyelinating disease with some semblance to the human disease, multiple sclerosis. So these coronaviruses have crossed species for so many years. 229E and L63 diverged about 1,000 years ago. And both of these are alpha coronaviruses, both cause the cold. OC43 by phylogenetic studies was shown to enter human populations 120 years ago. There's speculation that there was a major outbreak of respiratory disease at that point caused by OC43, but there's no evidence for it. And of course, MERS and SARS entered human populations more years ago than I have listed here. So this is a very brief slide just to show why I'm interested in coronaviruses. So this is something we did many, many years ago. And we were interested before there was SARS and MERS and these other, and COVID-19, we were interested in mouse hepatitis virus because it caused this demyelinating encephalomyelitis that I mentioned. And what this slide shows is if you look at the middle panel, this is a mouse that has no T cells or B cells, a RAG1 deficient mouse. And if you look in the third panel over you, the brown staining is virus, and this is the white matter of the spinal cord, and there's nice lines of virus-infected oligodendrocytes. And these are the cells that make myelin. And if you look in the white matter looks great. This is normal looking white matter, similar to what you see below in the mouse that has no infection, even though the color of the staining is different. Then if you add back T cells, you get destruction in 7 days. And so there's no virus, but now you have destruction. So this was one of the things that was interesting is, so how come these viruses cause a disease that the host is unable to eliminate without causing a lot of immunopathology? And this is a theme that, of course, is still true with COVID-19, but for all these human respiratory viruses, though it's not totally clear, but it appears like virus is being cleared at the time when people start getting sick. So it's the host immune response that causes disease. And we know for COVID-19, particularly as people get more and more sick, it's more and more the host immune response that in fact is controlled by steroids, which are being used in very sick patients, that seems to cause the substantial morbidity and mortality that we see. So I already mentioned, I didn't mention this, but this is a slide that has to be updated daily because tragically there's so many new cases every day. So now there's about 35 million cases. with a death rate of about 3.1%. This was actually the same rate roughly as it was at the end of January. Then it was about 2.8%. It went up, and the prediction was it was going to go down as we knew more and more about mild cases and subclinical cases. So it's gone down to 3%, but this is, hopefully it's really going to be down to way less than 1% once we get all the numbers in. And that's what some of the modeling shows. So, and this is a point, this is an old slide, but it just makes the point that the people who are older have the worst problems with this disease. And I think even more recently, it's more striking. I think the numbers are, if you're over 85, it's 20, this is a percentage of deaths, but if you're over 85, 20% of people die from the infection, and 70 to 80 years old, it's something like 10%. And if you're younger, the numbers are more like the top numbers here, 0.1%, 0.2%, which is why we have so much trouble actually dealing with the infection, because younger people think they're relatively resistant and may not take all the precautions that are required. So looking for animal models, because since we can't study this in humans, actually we can study it in humans, we just can't make interventions readily in humans, but we're certainly getting many more samples than we ever got from SARS or MERS and infected people. So we know that hamsters, macaques, ferrets, cats are all infectable. And I know some of these animals are going to be discussed later in the symposium. These animals all develop mild disease, and mice are resistant to SARS-CoV-2. And there are several approaches to making mice sensitive. Probably the thing that that people need to recognize, if they don't already, is that the reason that mice are not infectable is actually a 1 to 2 amino acid incompatibility between mouse ACE2 and SARS-CoV-2. So you can either change the virus by 1 or 2 amino acids, or you can change the mouse by 1 or 2 amino acids. So the ways that you can get to modify the mouse, in addition to doing what I just mentioned, is we also used a system of transduction of mice with adenovirus expressing human ACE2. As Matt mentioned in the introduction, we did the same thing for MERS. So this is a way to transduce any cell that's accessible to intranasally delivered adenovirus to infection. And it works very well, as I'll describe. There's ACE2 transgenic mice, which, depending on what the promoter is, have variable amounts of disease susceptibility. And then the third approach is to make ACE2, human ACE2 knock-in mice. This would involve either replacing the whole mouse ACE2 gene, or perhaps replacing just the 1 amino acid that you need to make a mouse susceptible. And this is actually the approach that we took, just replacing a single amino acid. And then the virus can be modified so that it can bind to mouse ACE2. So to start with the adenovirus system, this work has been published already, so I'll just go over it briefly. So the way you do this is we sensitize mice with an adenovirus expressing human ACE2, and then we wait 5 days so that if there's any inflammation from the adenovirus itself, it's mostly died down, and then we infect mice with SARS-CoV-2. And these adenovirus constructs are available from BEI. We also produce them at the University of Iowa, but I think BEI has greater capabilities now than we do. And when you transduce mice cells or the lungs with adenovirus 5 ACE2, you get the results shown here. If you look at the panel at the far left, you can see human ACE2 is expressed in tissue culture cells, and the third panel shows they sensitize these tissue culture cells for infection by the virus. And then if you look in the lungs, you can see that human ACE2 is expressed throughout the lungs. This is an area showing alveolar transduction. And when you infect these mice with virus, there's some weight loss, which is actually more substantial than we saw with MERS. But virus is always cleared in these animals within a few days, and the mice never die. And if you look for viral antigen, you can see it's extensive throughout the lungs, actually the same place where the transduction occurred. And histology shows, and this one, the far right here shows it probably best, this extensive infiltrate into the alveoli and edema in the lungs. And then we can use the system to assess various factors that might be involved in virus clearance. And what we do here is these are mice, because the adenovirus can transduce any mouse, we can basically use any animal at all for our initial infection. So here we show that here we transduce interferon knockout mice, that's in the blue line, or interferon gamma knockout mice, which was the green line, or STAT1 knockout mice. And the thing that is noticeable looking at virus clearance in the next panel is that virus is not cleared in the absence of type 1 interferon. That's the purple, that's the blue line, not cleared well. But what's more striking is in the absence of STAT1 signaling, virus is cleared more slowly, but cleared a little better than the interferon knockout, but they lose much weight. And the thing that's striking about this is in the original SARS epidemic, when you looked at black 6 mice, the only mouse, the young black 6 mouse that was actually affected by infection with SARS-CoV were the STAT1 knockout mice. So it didn't matter if you didn't have interferon, it didn't matter if you didn't have T cells, mice seemed to do just fine. But if you knocked out STAT1, that's when mice became susceptible and died from the infection. That work was done in Ralph Barrick's lab. And then if you look here at the histology, you can see in the STAT1 knockout mice, there's a much more extensive cellular infiltration consistent with worse disease. And these mice are also useful for analyzing the T cell response. In the upper 2 panels, you can see what happens when you take out either CD4 cells, that's the blue line, or CD8 cells, which are the green line. In both cases, virus is cleared more slowly than compared to the red bars, but it's cleared eventually. And if you use a rag mouse, it's not really cleared efficiently at all. So there's still residual cells here that do some clearance. And this system is also useful for identifying T cell epitopes in these mice. This shows general pools of peptides. And these experiments are actually done in Guangzhou by my former postdoc and present collaborator, Jin Zhao. And this shows that in biopsy mice, CD8 T cells preferentially recognize S1, and CD4 cells recognize N. And this system is now being used, has been used to identify individual epitopes. And that work is still being considered for publication. And then looking at other kinds of interventions, if you use convalescent plasma, which is used frequently now in patients, though the efficacy is not proven, it's probably going to have an effect and be useful in some patients. But because of the way things have been done, we don't really have evidence that it's in a good controlled study that it's actually useful. But here, if we give convalescent plasma, these mice no longer lose weight. That's the red line. And virus is cleared more rapidly. That's the red bar here. While sera from or plasma from either a MERS patient or a SARS patient had no effect. And if you look at the histology, you see the same thing. The COVID-19 patient sera basically prevented all the infiltration edema that we saw in normal mice, or mice receiving normal sera. And the bottom panel just shows remdesivir is useful by the same as expected. It's delivered here prior to infection. There's much less weight loss. Virus is cleared much more quickly. That's the red bars in the middle panel. And histology shows little evidence of infiltration. So remdesivir is a useful antiviral drug. One of the challenges is that I think it's most useful when it's given prior to infection, but in the US, it's mostly given when people are hospitalized, where I think its efficacy is going to be less useful, because as the infection goes on and it's more host-derived, eliminating the virus will be good, but will not be curative. Okay, so the other thing I wanted to talk about is our ACE2 mice. And these are mice we developed for SARS-CoV. This was done in my colleague's lab, Paul McRae, and he's a pulmonologist, and he chose this particular promoter because it's expressed in epithelial cells. So we developed these mice in 2005 or 6, and the transgene in this mouse is expressed on chromosome 2. There's estimated to be 8 copies. If anyone's using these mice, you can either use the heterozygous or the homozygous versions. They both work equally well. And one thing that's really striking that I will come back to during the talk is that in SARS-CoV-2, you needed very, very little virus to kill the mouse. So in fact, we found that as little as 3 PFU, which is incredibly small, delivered into the lungs or delivered intranasally, would kill the animals. And this was mostly via brain disease. And these mice are available from Jackson Labs. And so when we infected these mice, and many, many groups did this at the same time that we did, because these mice have been given out in thousands already. So what we found was that if you look at a if you look in the upper right-hand panel and look for survival, you can see mice receiving 10 to the 5th all die. But more strikingly, mice receiving 10 to the 3rd don't die from the infection. And with SARS-CoV, this was an absolutely lethal dose of the virus. So this is really different than SARS. These mice are much less susceptible to disease. And when we went and looked at where the virus was, we see viruses at very, very high levels at day 2 in the lungs. And in fact, our studies on MERS and SARS, and not as carefully done with SARS-CoV-2, but virus titers reach maximal levels at day 1 to 2 after infection in the lungs, and then they slowly come down. That's shown here. But in the brain, some animals, they get a brain infection. So SARS-CoV, all mice got a brain infection. Here you can see there's some that really have no infection of the brain. And these mice are still dying. So it shows that in these animals, lung disease is really the most important cause of death. And looking in the lungs, there's maximal virus antigen everywhere at day 4, and it decreases a bit by day 6. And looking at histology, you can see there's in the same kinds of infiltrates that we saw in some of the other systems. Then you can also see there's lots of edema here. That's what these the asterisk shows in this area here. In these vessels shows accumulation of fluid. And one of the striking things here is we see thrombi in the lungs of the mice that are sick with COVID-19 virus. And this is important because this is a major feature now. We think of pathogenesis in people who have COVID-19. And then if you look in the liver is pretty much unaffected, but we still see these large thrombi. So again, mimicking what's occurring in people. We don't think there's any, or not substantial amounts of virus. Whether there's any virus is a question, but this is mostly an endothelial cell infection, just like in people, plus or minus virus, really unclear. And then if we look in the brains, we see a very curious phenomenon. So at day 4, we see no labeling in the brain, and by day 5 or 6, it's actually overwhelming. So as opposed to SARS-CoV-2, where there's some SARS-CoV where it entered the olfactory system, and here you can see that this is the mitral layer being labeled. In these animals, it appears like there's a mixture of entry via maybe the blood-brain barrier breakdown plus the olfactory bulb. This is still something that's under investigation. And once again, we see thrombi, this time in the brain. And then one of the things that was interesting to us is the anosmia. So a lot of people who have COVID-19, either with mild disease or severe disease, have lost their sense of smell or taste. And I personally know individuals who have said, I can't smell. They're absolutely not sick at all. And then 2 weeks later, they can again. They may not have been diagnosed, but I'm sure that they had COVID-19. So we wanted to look in the nasal cavity for the virus. And what you can see here is there's nice labeling here in the respiratory and olfactory epithelium. And that in some places where you have labeling of the virus is actually some cell death. This is shown more obviously in the below there, where this is at day 5, not day 2, where you can see sloughing of infected cells, and you can see that in the H&E stain as well. And this is a sinus, and there's labeling all along the lining of the sinus. And then when you look for labeling actually in the epithelium a little more closely, it's clear that there's labeling of the sustentacular cells, and that's these lines here. So these have the morphology traditionally of sustentacular cells. This is interesting because in people, these are the cells that express ACE2, the receptor for SARS-CoV-2, which is not expressed by olfactory sensory neurons and other neurons in the brain, as far as we know. So, but this is a transgenic mouse, so the dynamics in the brain may be slightly different, but I think it's striking here how much infection of sustentacular cells there are. And so then this made us think, we wonder if this could be a model for anosmia. And we did several tests to examine this. I'm only going to show one of the 3 that we used for the sake of time. But the first thing what we did here is we took male mice and we put them in a cage with either female dander or male dander. And if mice are normal, what they'll do is they'll go and sniff around in the female dander and ignore the male dander. And so you can see that that's the case here in the lower panel. Mice are infected and then put in this cage, or uninfected. The uninfected mice spend all their time with the female dander, much less time with the male dander. And then once they've been infected by day 2, the difference is smaller. And then by day 3, there's really no difference in the amount of time they spend with the male versus the female dander. And this is what's up here in the upper left is just a panel to show that these mice at this time have not lost a substantial amount of weight. So it's not like they're not at the phase where they're really sick. So then the other thing we wanted to do, and I'll come back to the anosmia in a minute, is we treated mice with convalescent plasma. And this is, again, to test efficacy. And what we could show is that this was a high titer, so 1 to 1500. But with this high titer, we only saw really good protection with undiluted serial. When it was down to 1 to 500, we only saw partial protection. And when we looked where the virus was in these animals, remarkably, in these animals, looking at the right here, the animals that still died, there's no virus in the brain. So these are mice in the purple received 1 to 9 dilution. This 1 to 3, one mouse had virus in the brain at lower levels, and the undiluted had none. But there was no difference with the treatment in terms of labeling of the different parts of the olfactory epithelium, that's OE, respiratory epithelium, or other areas associated with maxillary sinus, so other areas associated with the nasal pharyngeal cavity. So this convalescent plasma prevented brain disease, diluted it did not prevent sufficient lung disease, mice still died, and it did not at all touch respiratory olfactory epithelium infection. So what we did next is we wanted to know what happened to anosmia in these mice, and basically showing the same exact plot. Now you can see that at 2 and 3 and 4 days post-infection, there was no difference in the amount of virus, there's no difference in the ability of mice to find male versus female dander. And then what we plotted these mice as a preference index, which is a way of measuring, trying to compensate for any issues of malaise or poor mobility. And the goal here is it's the amount of time spent in male versus female dander. If it's at 0, the mice have anosmia. And so we did this with female mice too. I'm not going to show those data, but those mice had virtually complete anosmia. Male mice at day 3 post-infection or 4 days post-convalescent plasma had nearly complete anosmia. At other times, they had a hyposmia, so partial loss of the sense of smell. So I think that I'm going to skip just mention these other approaches, which is modifying the virus. As I mentioned earlier, just a single mutation was needed to make the virus be able to infect mice. And these mice have been passaged by the Bert lab and become more virulent. And we're doing similar kinds of approaches as well. And then there's also an approach of using knock-in mice, which are being developed by several groups, including ours. So with that, I think I will stop, and I'll thank the people in my lab who did this, were mostly Abhishek, who did the anosmia experiments, Roy Jin Sun, who's now in Guangzhou, and Jian Zhang. And so with that, I will stop and take any questions.
Okay, thank you very much, Dr. Perlman. To start it off, we have a question from Tom Lane. So what cell types are infected by SARS-2? What are the pro-inflammatory cytokines or chemokines expressed within brains? And finally, are you seeing any spinal cord pathology?
So the last one, we haven't looked for spinal cord pathology. I don't think we're going to see any, but we haven't looked for it. In terms of cytokines, we see multiple ones affected in the lungs. We haven't looked in the brains. We did look in the brains during the SARS epidemic, and I don't remember there being anything specifically interesting there. But I would need to go back and look at that paper to see if that's in fact true. But we haven't done that yet with the brain. In terms of the kinds of cells that are infected, in the lungs, it's mostly alveolar and airway epithelial cells. In the brain, it seemed our original study showed it was mostly neurons. And part of the reason for that is this transgene is expressed in neurons, but it's so because that's where the promoter has very, very low levels of activity, but does have some. But it's still curious because SARS, it's the same mouse, but SARS infected these, spread from the olfactory bulb and really infected many, many cells in the brain. SARS-CoV-2 does much, much less infection. And in fact, as mice get older and there's less blood-brain barrier passage, we see very, very little infection and very focal infection in the brain.
So another question, are similar studies being done on human subjects at this time or planned in the future?
What does that mean?
Yeah, let's see. So presumably, Yeah, we can skip to the next one, and perhaps they can expand on it. So do the thrombi in the brain affect motor neurons?
Yeah, so we don't know what the thrombi do. They're not very common, but it's just striking that we see them. And what we're trying to do now is figure out, are they occurring at sites of virus replication? Do we see inflammation at sites of that lack virus replication? That's one of the things about COVID-19, is that all these manifestations, these peripheral manifestations like heart disease, brain disease, and blood vessel disease, while there's been reports of virus being found there, some of the reports are iffy, and often no virus is found whatsoever. So it seems like the cytokine or inflammatory mediator molecule expression is what's causing some of these changes. This is really something we need to know to understand this disease better.
Mm-hmm. And then we have a question from Michael Buckmeyer. Have you seen any evidence of viral deletion mutants as seen in MHV in the course of infection?
No, we haven't seen that. And during this epidemic, as you know, the only changes that there's been many changes reported, and almost none of them are fixed into populations of SARS-CoV-2, except for this one D614G, and that's been heavily investigated and may affect transmission, but not pathogenesis as far as we know.
Mm-hmm. And let's see, so in really sick infected mice, K18 or AD5 transduced, which infiltrating cell types are predominant in the lungs?
Well, we mostly see macrophages and lymphocytes, and there's some neutrophils as well. So I think the infiltrate is particularly the adenovirus system, where the mice really don't get sick. We see the usual panoply of cells. With the SARS-CoV-2 H2, the K18 ACE2 mice, we again see the usual group of cells, macrophages and T cells. We think macrophages contribute to pathogenesis, but we haven't proven that yet.
Mm-hmm. So another question. So if the virus is cleared, why is the immune response killing the host?
Yeah, that's a question. So this is I think that's why I showed that slide of the spinal cord early on, because it seems as if for whatever reason the immune response seems to go into high gear and not be able to come out of it. But certainly for SARS-CoV-2, I think that it's really hard to question, because in people who are sick, it seems like virus isn't completely cleared. With SARS and MERS, it seemed like it was cleared and people were still getting sick. But I think that what's the answer is there may be a little bit of virus left, and then the immune system hasn't wound down quickly enough. And there's also so much host cell damage that even if you don't have inflammation, that's why people take such a long time to recover, because they have residual fibrosis and other damage. That's been reported that people in the hospital with COVID-19, maybe a third of them have neurological sequelae without actually probably having had brain infection. So this is really a key thing. Understanding this and preventing it would be a huge impact on the disease.
Mm-hmm. So the earlier human studies question was actually referring to the anosmia. So whether that's actively being studied in humans.
Oh, I think it is actively being studied for many reasons, but I don't think we know anything about it yet. I think that from the animal studies, including ours, I think the sustentacular cells being infected may be important. How that exactly is going to work is not clear, because those are not the cells that actually sense odorants, but they're next to them. So they could be damaging them by producing cytokines, by inhibiting ion channel function that they have. The sustentacular cells are really more, they're more than just supporting cells. They have many other functions as well. So, but in people, one of the things that's really striking was when we got the results with the female mice, we were puzzled, because females do better than males in terms of this disease. But it turns out when you go back to the few studies that have been done in humans, women do have more of a loss of sense of smell than men do. So that's consistent with what we're seeing in our mice.
Mm-hmm. And another question from Michael Buckmeyer. So are there any genetic clues to explain the occurrence of families with multiple deaths?
I think we're slowly learning this. There were reports in Science a few weeks ago, or maybe not even published yet, showing that people who have worse disease may have genetic defects in interferon expression. And I think, judging from SARS and MERS, several molecules were, several genetic defects were associated, but nothing proven because there were so few cases. In this disease, we may really come up with specific risk factors genetically, but I don't think we're there yet.
And so there's another question. So in CD4 and CD8 depleted mice, does viral clearance coincide with the regeneration of those T cells?
With the regeneration?
I guess.
Yeah. I don't, that's a good question. I don't think so, but I don't know, because usually it takes a little longer for them to come back, but we didn't measure that in this case.
Mm-hmm.
So,
and then is age associated with severe disease in the mouse models?
Could you say that again?
Is age associated with more severe disease in the mouse models?
Yeah, so in SARS, definitely. This was a remarkable change. So B6 mice were resistant to SARS, and then by the time they were older, they were quite sensitive. With SARS-CoV-2, we have our oldest mice are only about 6 months old, but we'll be investigating that. And we're thinking that there will be increased lung susceptibility and decreased brain susceptibility, but that's still speculative. We don't know for sure.
Mm-hmm. And so another question, what causes the formation of thrombi?
Well, that's another question that needs research. We think it's damage to the endothelium, whether from cytokines or the virus, that still needs to be sorted out. And then with that, you deposit complement and platelets, and then you end up with these thrombi. But this is still also a work in progress. But it's really interesting because it's so important for the human disease. As you know, initially people who had respiratory problems were ventilated, and that turned out to be a problem because their lungs were actually not as sick as the blood vessels carrying oxygen away from the lungs. So ventilation actually was a negative help. And so understanding thrombi would be critical.
Mm-hmm. And there was actually a couple of questions submitted before the talk. So one is pretty challenging, and we won't hold you to it, but the question is, when do you think this pandemic will end?
Oh, well, I think that what I would guess is that I think we're going to still have problems through the end of 2021. And at that point, we'll either hopefully have enough immunity or a vaccine that worked. The thing that I think about that's a worry, though, is that if this virus doesn't change its virulence, then even if you only have 20% of the population being susceptible, people who have risk factors, if they encounter the virus, will still have problems. And maybe therapies will get better so that they won't succumb to the infection. But unless it becomes more attenuated, it'll always be a risk to people who are older or people who have comorbidities like obesity or hypertension.
Mm-hmm. And then perhaps one last question, again, a more general one. So regarding the likely vaccine. So if testing positive for COVID-19 results in 3 months of immunity, presumably hypothetically, would it be the same duration for the vaccine? So what do you think a vaccine will look like in terms of its protection? Yeah, that's the thing that we've been thinking about a lot, because we know that for people who had SARS 15, 20 years ago, they still have antibody and T cell responses. They had severe disease. We know with MERS, you had mild disease. The immune response seems to wane. We know common cold, people can get reinfected a year later or even sooner. So we hope the vaccine will be more like severe disease without disease severity. But because the vaccines have been used no more than a couple of months, we have no idea about durability. Certainly from the MERS vaccines that were used using either the Oxford system or the DNA platform, these were done a few years ago. We know that immunity to each vaccine is different, so they're not all the same. One vaccine may give a better T cell response, another may give a better antibody response. That's why some vaccines require 2 injections instead of just 1. So, and how long they're going to last, that's the major question. I don't think it'll be 3 months. I think it'll be nearer to a year. I think even the reports of 3 months probably underestimate how long we're really going to have protective immunity. But if it's like the cold, at a year, we may need reboosting. Yep. Okay. Well, thank you so much. We're out of time, but thank you for a very fascinating talk. And so, yeah, with that, I would like to introduce the next moderator, who's Dr. Dawn Forthall. Dr. Forthall is a professor of medicine and molecular biology and biochemistry, and chief of infectious diseases in the UC Irvine Department of Medicine. So welcome, Dr. Forthall.
Thanks very much, Matt. It's my pleasure to introduce our next speaker, Dr. Emmie DeWitt. Dr. DeWitt is currently the chief of the molecular pathogenesis unit in the Laboratory of Virology at the NIH's facility in Hamilton, Montana. She obtained her PhD in virology in 2006 from Erasmus University in Rotterdam, the Netherlands. She focused there on the pathogenesis, replication, and transmission of influenza A virus. But she developed an interest in working under BSL-4 conditions, which brought her to Hamilton, where they have such a facility, in 2009. And while there, her research first concentrated on Nipah virus, as well as MERS and the 1918 influenza A strain. More recently on Ebola virus, and her interest in that led her to leadership of Ebola virus diagnostics in treatment units in Monrovia, Liberia during the big Ebola outbreak in West Africa. And now, of course, with the emergence of COVID-19, she's moved to the forefront in developing SARS-CoV-2 animal models and using them to test drugs and other measures to manage the pandemic. So many, many thanks to Dr. DeWitt for taking the time to talk to us today. The title of her presentation is Non-Human Primate Models for the Development of COVID-19 Countermeasures.
Thank you so much. Let me try sharing my screen. Yeah, let's see if that works. All right, that should do it. Yeah, so as is clear from the introduction, I'm really interested in viruses, emerging zoonotic viruses that cause respiratory disease. So usually when there's an outbreak, I get interested and start working on it. And that's how we jumped on SARS-CoV-2 when it emerged. Of course, although we've always been talking about, oh, these things can cause pandemics, I also never thought that we would be in the situation that we are in now. But we kind of, thanks to our work on MERS coronavirus previously, we could really hit the ground running and make some contributions, I think, that were pretty important, at least for getting some treatments and vaccines into clinical trials. So my talk today will really focus on our work in non-human primates here at Rocky Mountain Labs. So I wanted to highlight a little bit because, of course, we get a lot of questions, why in this day and age do we still need animal models? And I think when a new virus emerged, they are still really essential for our understanding and our research about these new pathogens. So first of all, we need to fulfill Koch's postulates. So we need to show that the agent that was isolated from the patients with the disease actually causes that disease when you infect animals, to show that you've really identified the causative agent of the disease. But they are also really important to understand what's really going on when people are sick. Of course, it's really hard to take a lot of repeated sampling. You have always the problem that you don't really know when and how people were infected. But in animals, of course, you can do really controlled experiments that will allow you to get really detailed information on how the disease progresses. And you can do this under ideal circumstances with regard to how and when you can collect and process your samples. Of course, they really are an essential part of preclinical development of any countermeasure. So vaccines and antivirals, almost all of them, regulatory agencies will insist on seeing efficacy data in animals before they will allow clinical trials to proceed. So we are very well equipped, unlike a lot of labs, to do non-human primate studies at BSL 3 and 4. And so this is one of the animal models that we usually focus on. That's also, of course, one of the animal models where we get a lot of questions about why do we still need to do animal experiments in non-human primates. And it's really simple. Sometimes they are the only natural infection model. When you think, for example, about MERS coronavirus, originally the only animals that we were able to infect with a wild type virus were non-human primates. And so when you think about SARS-CoV-2, of course, we had information from MERS, but also more importantly from SARS that uses the same receptor, where actually most of the other models that were available didn't really show any disease. So that really led us in the direction of non-human primates. The non-human primates, of course, are genetically closer to humans, which we sometimes think would help us get the disease that's closer to what we see in humans, but of course that's not always the case. But that would also allow you to test host-directed therapies better, just because the host is more similar. And then sometimes compounds that you think might be interesting to test as antivirals don't really work in other animal models. And we have some experience, and I'll show our data on remdesivir, where remdesivir is metabolized differently in mice than in humans, and that makes it very hard to test the treatment efficacy in a rodent model, whereas it's metabolized in the same way in non-human primates. So as soon as we managed to get a virus isolate, we tried to develop a non-human primate model in rhesus macaques. But what's important to realize is that this work was done back in February. So back in February, we didn't know everything about COVID-19 that we know now. And actually, thinking back, our knowledge was pretty limited. So we knew that it causes respiratory disease and that some people got really mild disease and then other people got a fatal pneumonia. We knew that the people that got severe disease would usually progress in their second week. For a while, the pulmonary infiltrates, especially on the CT scan, were used as a diagnostic method. So those are really a hallmark of the disease, or at least at the time. And there were some data available that in people you could take swabs or samples from the lower respiratory tract samples, and they would contain viral RNA. So this is really what we were trying to model. We were just trying to get a model where we would see respiratory disease. Of course, preferably we would see the severe disease, because that just makes it a lot easier to test a lot of the countermeasures. But this is kind of what we were aiming for. So the way we did our experiments was we started a study with 8 adult rhesus macaques, 4 males and 4 females. And the way we set up the study was that we would have useful data no matter what happened, whether the animals got sick or not. We were trying to get a lot of data out. So we had a group that we just monitored to see if they would get disease, and then we would follow those out to 21 days. Of course, if they would get very sick before, they would be euthanized, but if they wouldn't get sick, then we would euthanize at day 21 so that we could see if they would seroconvert. And then we had a group that we euthanized on day 3 after inoculation, because we know that really these viruses, usually their replication peaks really early in the respiratory tract. So if you wait too long for the animals to develop disease, you might actually miss that peak of replication. And so we decided to also take one group on day 3. So in between, we sampled these animals a lot. We collect swabs, we take x-rays. On the animals that were allowed to progress out to day 21, we also collected bronchoalveolar lavages. And then for our inoculation, we use a combination of 4 routes: intratracheal, intranasal, oral, and ocular. And that's because that way we target the upper and lower respiratory tract directly, as well as the mucosal surfaces that might be involved in real life transmission of the virus. So this is what happened. Every day after we inoculate the animals with the virus, we have the same person checking the animals, and they score the animals for several categories, like do they have respiratory signs, do they still eat, are they active, do they groom, all of those different things then add up to what we call the clinical score. A clinical score of 35 is our humane endpoint, so you can see these animals get sick, but they don't get severe disease. And these scores really are made up of a combination of the animals no longer being interested in food, but also respiratory signs. And then on our x-rays, here you can see an example of one animal over time where we saw these really clear infiltrates in these red circles. And as you can see, it starts to clear by day 7. So that's also what we see here. The animals really start to recover about a week into the infection. And then some animals recover a little bit faster, but after about 2 weeks, they're all sort of back to normal. We also have our veterinarians score the infiltrates that they see on x-rays. So that adds up to this score so that you can actually sort of quantify what we see on an x-ray, which is important later on when you start to use the model for testing of countermeasures. So really these infiltrates, we were really happy to see, because at the time we did these experiments, that was such an important part of the human disease. That made us really feel that this would be a good model to do some more work in. So we took a lot of swabs, like I said, and actually there, the shedding pattern that we saw was fairly similar as well to what was seen in humans at the time. So we see high shedding early on from the nose that slowly declines over time. And in the throat, we see more intermittent shedding. And then there's here just really, and this is what we see almost every time we do an experiment, that there is one animal where the rectal swabs will also come up positive for a period of time. We've never seen any virus in the urogenital swabs or here in urine, and also the animals are not viremic at any time during the study. As a sample of the lower respiratory tract, we did bronchoalveolar lavages. And as you can see, we see very high amount of viral RNA in these lavages, and also early after infection, we see a high amount of infectious virus. So really what it looks like based on these data is that the virus replicates really quickly when we inoculate the animals. Probably the peak of replication is within a day or two after inoculation. So here are the data for the animals that we euthanized on day 3. So in all of the lung lobes, so we test all of the individual lung lobes. They all have a high amount of viral RNA, and we can also isolate virus. And then in the rest of the respiratory tract, it's more, it's not as clear. Some animals will have viral RNA and some won't. But there is some replication going on in the rest of the respiratory tract. And then other than that, really, we don't see anything in the brain. We see a little bit here in some of the intestinal tract tissues, but no real evidence of virus replication, also not in histology. So it's kind of really confined to the respiratory tract in non-human primates. When we look at histology, we see what I would call classic multifocal interstitial pneumonia. That's just mild to moderate. It's not severe, which matches up with what we see clinically in the animals. So here, this is one of the part of the lung lobe of one of the macaques on day 3. And here you see this area is affected by a lesion, whereas this area is still normal. And when you look at these lesions more closely, of course, at the time we did these experiments, there wasn't a lot of data from human autopsies, which are also really hard to compare because in humans, of course, you're always looking at someone who just passed away, and they have been on a ventilator often. So it's we can see the similar features, but it wouldn't look exactly the same. We see in these lungs, when you look at these lesions, you see here a vessel surrounded by this huge infiltrate of inflammatory cells. We see some edema, and then here we see formation of hyaline membranes, which were also a hallmark of SARS, the first SARS. And so when we saw this, even though there weren't any data from human autopsies, this made us feel like this would probably be what you would see in humans if you had the data, because this is what we classically see in these animal models of coronavirus. And then here, by immunohistochemistry, we could detect viral antigen in macrophages and type 1 and 2 pneumocytes. We also looked by EM, and this is one picture that's really nice because it really replicates what we see here in the histology, except it's a little harder to see what you're looking for. So here you see a very thin cell, which is a pneumocyte, and here as well. And in between, the interstitium has expanded with a lot of edema and fibrin, and actually also some inflammatory cells as well. So it really captures that same picture, but then additionally, where you see the arrowheads, we see a lot of budding virus particles, showing that the virus is really actively replicating in this pneumocyte. So like I said, we had several reasons to think that this was a really good model, and it would at least, although the disease was not severe, it recapitulated some of the essential parts of the disease in humans. And so the first thing we did after that was to test the efficacy of remdesivir. I don't think I have to give much of an introduction on that anymore. It's a nucleotide prodrug that was developed by Gilead. We had done some work with remdesivir, and that's why it was first on our list, to show the efficacy of remdesivir treatment for MERS coronavirus. And that actually looked quite promising, and that's what made us think that this would potentially be a good drug to use in human patients as well. There were already quite some safety data, and while as we were starting the animal experiments, they were also getting ready to start clinical trials in patients with COVID-19. So we used the same setup for our study, and here's an overview. So we really we end the studies for these treatment studies on day 7, and that's because on day 7 we are just after the peak of replication, and you will still have all the histological changes. So you kind of have, although it's after the peak of replication, you can still detect the virus, but you can also detect all of the histological changes that, of course, will tell you whether animals are developing a pneumonia. So we had 2 groups of 6 animals, one of which received remdesivir, which is an intravenous treatment. So we would administer that every day intravenously in the animals. Clearly here you can see that the control animals in black, they are getting sick, but the animals that received remdesivir really are hardly getting sick. And what you see here is really just the animals not eating as well as they normally would, which could be from the SARS-CoV-2 infection, but it could also be because they have to be anesthetized every day to get their IV treatment. Similarly, there was some infiltrates visible on X-ray, but they were definitely a lot less with remdesivir treatment than in the controls. Here also we did bronchoalveolar lavages on day 1, 3, and 7. So when you look at viral loads, there's a small difference, but when you look at infectious virus titers on day 1, which is 12 hours after the first treatment, we already see really a significant reduction in the infectious virus that's in the bowel, which is hopefully reflecting that the virus isn't replicating in the lung. Where we did not see an effect was in any of our swabs, and I'm only showing the viral RNA, but this is true also for infectious virus titer. So really, if you treat the animals with remdesivir, it seems like the virus isn't replicating in the lungs as well, but the animals are shedding the same amount of virus, which of course means that if you translate this to human, they might still transmit the virus. So when we did necropsies on day 7, here you can see that there's a lower viral load in the lungs in animals treated with remdesivir. And actually, we couldn't isolate any infectious virus from the lungs of the remdesivir treated animals, whereas we could isolate virus in 4 out of 6 of the vehicle treated animals. Here, I think, is the most important part. So obviously, in the remdesivir animals, we see these tiny areas of inflammation that really are very subpleural, and they are very minimal, and they're not present in all of the animals that we treated. Whereas in the control animals, we see that same typical lesion. So really, although the animals are shedding virus from the upper respiratory tract, they are protected from developing that pneumonia. And so I think it's really important to realize, Dr. Perlman already mentioned it in the previous talk, but here we show that if you treat early enough, you can really prevent people from progressing to disease. And I think, I guess, what we should be doing is trying to find a way to treat people earlier in the disease rather than wait until they are hospitalized, when you know you're already past that peak of replication and the effect is very minimum. So these data, together with some of the clinical trial data, of course, went to the FDA, which led to emergency use authorization, and the same was done in Europe. So I think that's one great achievement that we managed to get during this pandemic really early on. But of course, now even there's a very famous patient taking remdesivir. So we, I guess we contributed to that as well. So of course, vaccines came up. They are probably going to be a more essential part of controlling the pandemic than the antivirals. So we've been working on this ChAdOx. So it's a chimpanzee-derived adenovirus. It's replication defective. Already before this pandemic, phase 1 clinical trials had been done with other ChAdOx. So the same vector, but different vaccine antigen. And so immediately in January, the Jenner Institute started making a ChAdOx that expresses the SARS-2 spike. And then here we tested it in non-human primates. These are the data, the serology data. So as you can see here already, so there's a group that only got a prime in red, and then there's a prime boost group in blue. So you can see if you give prime only, you do get antibody response. This is just IgG, but here also you get a neutralizing response, and that is higher if you give a boost as well. You can see then when we challenge the animals, these are the viral loads in the lung. So we use genomic RNA as well as subgenomic RNA. And as you can see, really, the virus replication in the lung is greatly reduced in the vaccinated animals, regardless of whether they received a prime or a prime boost vaccination scheme. Again, here we see that the shedding isn't really reduced in these vaccinated animals. And of course, that is a bit of a concern, and it's something that we need to look into to see if we can achieve better protection in the upper respiratory tract, but this is probably going to be very difficult. So for now, it looks like this vaccine would mostly protect against replication in the lung, and that's what we see in histology as well, where the vaccinated animals have hardly any changes histologically in the lungs, but the controls again show that typical interstitial pneumonia. So these data, really the day we reported these data, they start the next day they started the phase 1 clinical trial. Of course, by now the data from that are already available, and thousands of people are enrolled in the phase 3 trial already. So I hope I still have some time, because I wanted to put this in perspective and show some of our newer data, because of course, like I've shown, the rhesus macaque model actually works quite well when you're testing direct acting antivirals or vaccines, because we can look at reduction in virus replication and pneumonia. But because we're not getting that severe disease, we can't really test any of the host-directed therapies in this model. And that's definitely a limitation. And also probably because we don't get severe disease, we don't see a lot of the non-respiratory issues like the thrombi that were just brought up. So the clotting complications, the CNS involvement, we don't really see that. So if you can't see it, you can't figure out how this is developing and what you could do about it potentially. One way to potentially deal with that is, of course, to start modeling some of the comorbidities. And that's one thing we are trying to do, but it's not what I wanted to talk about. One thing that we tried was use a different non-human primate model, which is African green monkeys. And as was mentioned earlier on, I have a big research interest in Nipah virus as well. And Nipah virus actually causes acute respiratory distress syndrome in the African green monkeys. And so we were hoping that there was something in their genetic background that would make them more prone to developing acute respiratory distress. And so maybe they would be a more severe model. And so we basically did the same experiment that we did originally for the rhesus macaques, except we now added 2 controls that we inoculated with irradiated virus. So the irradiated virus is non-infectious. And we use these as a control. For one, we often get a lot of questions about, you're putting in a lot of virus, so the virus that's coming out is probably not replicating, it's just remaining inoculum. But also we wanted to do some single cell sequencing. And for that, we needed a good control. And the irradiated virus is kind of the closest control that we could think of. Other than that, we did the same experiment. So as you can see here, the animals do all get some clinical scores. So here in this blue color are the 2 animals that were inoculated with the irradiated inoculum, and you see they get a score. And that's really because they have been anesthetized regularly and they get intubated to do the intratracheal inoculation. These animals show some respiratory signs. There's one animal that sticks out here, and that animal actually is not showing more severe respiratory signs. It's actually showing a complete lack of appetite. So this animal went completely off food for a while. That's why the scores are higher. And I'll come back to that in a little bit. So I'm going to skip some of the data that I showed for the rhesus macaques. I just wanted to show here we're scoring the gross lesions. So you can already see that these animals do have gross lesions on the lungs, meaning they are developing a pneumonia. We did look at shedding. When you, at a first glance, this looks pretty similar to the rhesus macaques, actually, with this high shedding early on in the nose that then goes down, intermittent shedding. And then here there's one animal where the rectal swabs are positive. And these squares indicate that it's the same animal that didn't eat for a while that I talked about in the previous slide. There is a problem with these data, actually, because here you can see the animals that are inoculated with the irradiated inoculum actually are coming up positive in the nasal swabs, even still on day 3, indicating that the RNA that is in that irradiated inoculum is actually pretty stable. And so this is not a very good measurement then of what is actually going on and whether virus is replicating or not. And so we decided to implement testing of subgenomic RNA, which should only be present if the virus is actually replicating. And you can see here that we're not finding this in these irradiated inoculum animals, even though it is present in the inoculum. And I'm getting a lot of questions about that. It's mostly people who are not virologists, I think. But of course, when you grow a virus stock, the cells are lysing. So the subgenomic RNA is going to end up in the stock as well, even though there's no replication in the media itself. But now you can also see what is actually quite nice is that we do get replication really going on in the upper respiratory tract. But after about a week in, it's really all coming back negative. And this is, of course, important. Earlier on, there were a lot of stories about people who kept shedding RNA forever. And that's kind of what we see here, where 3 or 4 animals are still positive on day 10. But when you look at subgenomic RNA, they are not. Now, when we compare that to infectious virus, there's a lot less infectious virus that we detect. But of course, the question is that because it's not there, or is it because our cell culture system is not sensitive enough? And it's probably a little bit of both. So I think the subgenomic RNA really is a good measurement for virus replication, a little bit better than looking at infectious virus, but definitely more informative than just the genomic RNA alone. And we also looked at the bowel fluid to look at replication in the lower respiratory tract. And again, here you see high viral loads in the bowel fluid. When you look at genomic RNA, again, one of the animals that received irradiated inoculum is coming up positive, but you're losing those animals when you just look at subgenomic RNA. So these are the viral loads in the lungs at necropsy on day 3 versus 10. Again, a lot of replication going on in the lower respiratory tract and a peak of virus replication early on after inoculation. And again, although the lesions were somewhat less severe, we do again see that interstitial pneumonia in the African greens as well. So unfortunately, they are not a better model than the rhesus macaques. They did not develop severe disease, but I think they are also a model that you could use alternatively to the rhesus macaques if for some reason you would prefer to use African greens. I don't think if you're doing antiviral testing that it would matter much. Here, one more interesting thing is that animal that didn't eat and that then had that positive rectal swab over time is the only animal we've seen so far where there's actually virus replicating in the ileum. So here the epithelial cells are positive for virus antigen in this animal, which might, of course, we're just assuming that this might be the reason why it was not eating well, that this was a discomfort to the animal. So I will really briefly go over only a tiny section of the single cell sequencing analysis that we did on the lungs. We actually also did draining lymph nodes. So I would say if you're interested, go check out our preprint. So we managed on the lung sections to really divide this into all of the different cell types that should be there. And we used different marker sets to confirm that these cell types were actually those cell types. And then, of course, the nice thing about that is that you can see where can we actually detect the viral RNA. And you can see that really the largest, the cell type with the largest proportion of viral RNA is the macrophages. But of course, macrophages are cleaning also up all the infected cells. So one question that we had was, is the virus actually replicating in these cells or not? And one way we tried to figure that out was to look at the different transcripts. Of course, we use the 10x genomic system, so there's a 3' bias to the transcripts that we're going to find. And so all of these are now relative to n, which is really close to the 3' end relatively. And all of these cells kind of look the same, except here, when you look at the pneumocytes, they have this very high amount of ORF1ab transcripts, and these are actually some of the non-canonical subgenomic RNAs that we are detecting. And so this makes us think that really, although we see a lot of macrophages positive, that the infectious virus is probably really coming from the pneumocytes rather than from the macrophages. This also, even though we see viral antigen in immunohistochemistry, we actually don't see viral RNA much in the macrophages when we do in situ hybridization. We really only detect it in the pneumocytes. So that kind of confirms this. We looked at I kind of lost my train of thought. Anyway, this is all I really want to say. If you want to read more about what those macrophages are doing, then we have some nice analyses in our preprint. And so I think I'm probably a little over my allocated time already. So I'll just say that doing these studies requires a lot of help. And on this slide, you can see all of the people that have been instrumental in getting this done. And with that, I'd be happy to take questions.
Thank you very much, Dr. DeWitt. That was terrific. Really appreciate your presentation. We only have time for a couple of questions. The first one comes from Dr. Messaoudi, who asks if you've attempted to infect the macaques of Indian origin, and if you have or haven't, whether you might see differences in disease expression.
Yeah, so we have, and this is really a pure coincidence, because when we normally use Indian origin rhesus, and this is what we had in our facility when we got the virus, which is why we use the Chinese. So ever since these first two experiments, we've been using the Indian origin, and it looks like we see a little less disease. We don't see a difference so much in virus replication. But we do see fewer gross lesions in the Indian origin rhesus. It's still enough to do vaccines testing, but there seems to be a small difference.
And I had a question for you. The monkeys are, of course, outbred, and it brings up the question whether in a disease that in humans generally expresses itself as very mild, whether you really need to do hundreds and hundreds of monkeys to start seeing severe pathology. In other words, it may not be that there isn't any, it's just that you haven't infected the right animals.
Yeah.
Yet.
that's something that we were thinking about too early on. I wouldn't say that we have done hundreds of animals at the moment, but we have done, of course, quite a few vaccine studies and other antiviral studies, and we haven't seen any animals develop severe disease so far.
One other question, have you tried older and or diabetic monkeys?
Yeah, we haven't tried diabetic monkeys, but we have tried older monkeys, and actually the older monkeys are also obese. So those things often go together. There, I would say we're still analyzing the data. The disease is not necessarily more severe, but the recovery is slower in the older animals.
Great. Okay, I think that's all the time we have. So thank you once more, Dr. DeWitt. We really appreciate you taking the time and talking with us. Now I'm going to turn over the mic to Roberto Tinoco, who's a faculty member in the Department of Molecular Biology and Biochemistry.
Thank you, Dr. Forthall. Good morning. Good morning, everyone. It is my pleasure today to introduce to you Dr. Ilhem Messaoudi. Dr. Ilhem Messaoudi is an expert in immunology and virology. She is a professor of molecular biology and biochemistry here at UCI and is a member of the UCI Center for Virus Research. She received her Bachelor of Science in Biochemistry from Lafayette College and her PhD in Immunology from Cornell University's Graduate School of Medical Sciences and Memorial Sloan Kettering Cancer Center. Dr. Messaoudi's work on varicella zoster virus, which causes chickenpox and shingles, Ebola, and SARS-CoV-2, is recognized by her numerous grants and publications in the area of viral immunology. She studies immune responses in vulnerable populations, such as those in pregnant mothers and in aged and obese individuals. Today, she will present her latest research on our understanding of how SARS-CoV-2 immune responses in aged and young is impacted in patients. With that, I would like to welcome Dr. Messaoudi.
Thank you, Roberto, for that kind introduction. And thank you, Bert and Sanghyuk, for inviting me to present in this wonderful symposium. So I'm going to go ahead and share my slides. And as Roberto indicated today, I'm going to be sharing with you unpublished results that culminated after several months of really hard work by my lab and with huge assistance from the ICTS here at UCI, as well as the COVID-19 biospecimen repository. And so we're going to talk about longitudinal profiling of peripheral blood immune responses to SARS-CoV-2 in young and aged patients with varying disease severity. So I think Dr. Perlman did a great job, and I don't want to belabor the point, but as we all know, COVID-19 is a respiratory disease caused by novel coronavirus, SARS-CoV-2. We know that this virus causes a wide range of symptoms that can go from asymptomatic disease. We are estimating that maybe 40% of the cases are asymptomatic, mild disease that can be characterized by fever, cough, myalgia. And then in some individuals, disease can progress to take on a very severe form that is associated with acute respiratory distress syndrome, endothelial barrier dysfunction, and coagulopathy. And the risk factors for developing the severe disease include age, body mass index, and metabolic disease such as diabetes, cardiovascular disease, and hypertension, as well as male, sex, and potentially blood group types. So I just want to have a very, very brief introduction to the immune response to viral infections in the respiratory tract so that we can set up the stage for the rest of my talk. So probably as many of you remember from their immunology classes, the immune response in the respiratory tract begins the moment that a virus infects the respiratory epithelium. The intrinsic defenses of the respiratory epithelium, which consists primarily of producing copious amounts of type 1 interferons and chemokines, lead to the quick recruitment of innate immune cells, notably dendritic cells, alveolar macrophages, and NK cells. And the coordinated action of this innate immune response does two things. One is suppresses viral replication and creates antiviral states and bystander cells, but also contributes to the recruitment of the adaptive immune response to the site of infection. So the innate immune response buys time for antigen presentation to occur by dendritic cells to T cells to initiate the development of both CD4 and CD8 T cell responses, as well as B cell responses in the form of antibodies. And this entire response has to be very tightly regulated so as to maximize viral clearance, but minimize the damage to the host tissue. And whenever we have a disbalance in this immune response is usually when we see a lot of pathology. And so this is the case with SARS-CoV-2. The initial stages of the infection are very much like we understand from other respiratory viruses. It infects the respiratory epithelium and leads to the generation of robust type 1 interferon, as well as several inflammatory cytokines, and the recruitment of both dendritic cells, alveolar macrophages, and NK cells. But at this, there's a bifurcation, and the immune response can either quickly control viral replication, minimize tissue damage, and that leads to the quick resolution of disease, or there's an over-exuberant immune response that ultimately leads to the development of vascular leakage, edema in the lungs, which leads to pneumonia, and a dysfunctional immune response due to the presence of copious amounts of type 1 interferon and other inflammatory factors that at this point become detrimental rather than helpful to the immune response. And so there's been a number of wonderful studies that have come out recently describing the immune response following SARS-CoV-2 infection. And so there's several clear take-home messages from these studies, including that aberrant inflammatory responses and hyperinflammation is associated or correlated with disease severity. So this is the concept of the cytokine storm. So the more cytokines and the higher levels of cytokines, the worse the disease outcome is. There are reports indicating that T cell numbers may be reduced, so there may be signs of lymphopenia in the patients with severe disease, and that T cell responses may or may not be dampened in severe disease. So there's still several, different reports depending on what type of T cell assays are being used to measure antigen-specific or just bystander T cell activation. And then we know that plasma B cells are detected early. We know that antibody responses can be robust, and that the strength of the antibody response positively correlates with disease severity. But as Dr. Perlman said earlier, we still, the jury's still out in terms of what these antibodies are doing, and we still need more data from the convalescent plasma studies to really get a good understanding of what antibodies are doing. So there's a few limitations to some of the studies that are out there. Most of them don't consider days post-symptom onset. Some have big differences in the definition of disease severity and categories. And then some of the studies really very acutely focused on the severe disease, but do not include mild and asymptomatic individuals of varying ages, making some of these key comparisons very difficult. And so we attempted to address some of these outstanding questions, and we really had two questions at the onset of the study. One is to try and understand the longitudinal changes in immune cell subsets in response to SARS-CoV-2. And then how do these immune responses correlate with clinical disease outcomes? And the data I'll be showing you today comes from a collection of, sorry, these numbers are actually just switched, I just realized. So we actually have 36 healthy donors, 12 mild/asymptomatic COVID-19 patients, and 46 patients who are hospitalized with severe COVID-19 infection. And the severity of their disease ranges from just being on the ward to being intubated in the ICU. And we have a handful of patients in our cohort who unfortunately did succumb to disease. We collected blood specimens from these subjects. We isolated plasma and PBMCs. The plasma was used to measure antibody levels as well as levels of immune mediators that are in circulation. And the PBMCs were used to carry out a number of flow cytometric experiments as well as single cell RNA-seq experiments. So I'm hoping to get through most of the data today. We'll see how we do on time. And so the cohort characteristics is as follows. So patients range in disease severity from, like I said, unfortunately death all the way to convalescent or healthy controls. There is a bias towards male gender in our severe cases, and there is an increase in age in the categories with really severe disease. Because of where we're located, our patients are mostly Hispanic. And I really, this sample collection would not have been possible if it wasn't for two individuals who really made our access possible, and that is Dr. Dan Cooper and Dr. Frank Zaldivar from the ICTS, and Dr. Rob Edwards and Dr. Trivia from the pathology lab, who worked tirelessly to actually collect for us discarded specimens from either the various remdesivir trials or from the CBCs that were being done on these patients to make it possible for our lab to have access to these samples. Okay, so just briefly, I wanted to show that although when we take all the three categories and compare them to one another, we don't see any differences in the distribution of age. However, if we look within the severe category, as well as the asymptomatic individuals and look at their ages, and we can see that the group of individuals who did succumb to disease are older than all the other categories. Also, as reported by some other studies, although the number of white blood cells in circulation is not different, we do see a pretty significant lymphopenia in the individuals who do end up getting hospitalized with COVID-19. And so I will revisit this question of lymphopenia in the next few slides. Okay, so I'm going to start off by looking at antibody responses. And right here, this is a principal component analysis looking at the distribution of antibody responses directed against either NP, nucleoprotein, or RBD, so the receptor binding domain of the spike protein. And if you follow here, you could see that we have a number of samples that we call the pre-COVID samples. So these are plasma samples that were collected in 2018 or earlier, and so they're negative. So that just gives you a sense of where negative samples should cluster, and they cluster very nicely together. In purple are convalescent samples, and you can see that they're quite distinct from the plasma samples that were collected pre-COVID. And then the cyan color is individuals who are asymptomatic. So who are these asymptomatic individuals? They're people who either had mild disease, showed up to the ER, got a sample taken, but then ended up being discharged home because they did not require admission to the hospital, or people who showed up for elective surgery and then found out that they were actually COVID positive and then had to go home. But what I wanted to kind of draw your attention to is the fact that it actually, from this principal component analysis, looked to us like our asymptomatic individuals had potentially a more robust antibody response than severe patients with severe COVID-19 during their days post symptom onset 1 through 5, which is here in red. However, as the disease progresses, we see the antibody titers increase. So the dark green is days post-symptom onset 6 through 10. Then this yellow graph is 11 through 15. And this pink graph is anything greater than day 15. And so to look this a little bit different, we can look at the distribution of the strength of the antibody, binding antibodies. And this is depicted in a heat map. We can see that relative to what we call the pre-COVID plasma samples, asymptomatic individuals have a significantly greater abundance of binding antibodies to both NP and RBD, but that indeed individuals who had severe COVID-19 on days post-admission 1 through 5 actually have indistinguishable antibody levels compared to the pre-COVID samples and lower than those who are asymptomatic. But then the antibody response quickly increases as disease progresses. And so we decided to take a closer look at that and ask if all hospitalized patients showed up with low antibody titers, or was that dependent on disease severity. And so when we look here at either IgM levels or IgG levels to NP, we can see that individuals who eventually ended up succumbing to disease in that early phase of days post-symptom onset 1 through 5, they have a significantly lower levels of IgG against NP and IgM and IgG against RBD. Now they quickly catch up to the other categories, but it's potentially really, really critical to have antibodies in this early phase as all those individuals ended up succumbing to disease. And so we took a look using flow cytometry at the robustness of B cell proliferation using Ki67 as a marker for a cell cycle. And so this is just using flow cytometry and looking at circulating B cells in the blood. We did not look at plasma B cells, which is something that we're very interested in doing, and we will go back and look at our samples more closely with that. But just looking at circulating B cells, what we were struck by is the fact that there is a higher frequency of marginal zone-like B cells, and these are B cells who are transitioning from naive to memory and are capable of making IgG as well as IgM. And the levels of those B cells were higher in individuals who either had mild or had asymptomatic disease. And similarly, B cell proliferation in all categories, whether it's looking at total B cells or naive B cells or marginal zone-like B cells, were all higher in individuals with asymptomatic or mild conditions. So this tells us that maybe individuals who do have mild disease have a jump start on making antibodies and getting B cells to proliferate and isotype class switch compared to those who end up developing severe disease. So the next thing that we looked at was the changes in circulating immune mediators, and we did this using Luminex. And again, this is kind of all the data, 45 analytes compressed into one principal component analysis. These are our healthy controls, and you can see that they cluster very nicely together and cluster away from all the samples collected from those individuals infected with SARS-CoV-2. We can also see that the profile of asymptomatic individuals or those with mild disease is quite different from that of patients who end up being hospitalized, and that the profile of these immune mediators does change with days post-symptom onset. And so to take a quick look at these mediators, and I know there's a lot of data in this bubble plot and it's a lot to go through, but the first thing that I wanted to bring to your attention is even though these individuals in this category of mild/asymptomatic, most of them did not know that they had SARS-CoV-2 or just had mild symptoms, that they still have significant changes in the levels of several immune factors that are important for antiviral immunity, including, for example, type 1 interferon down here. And so that's really important, and especially type 1 and interferon alpha, their levels are actually pretty high and commiserate with what you would see in patients with severe disease. But other things like interferon gamma and IL-12, IL-15, IL-17 responses were all quite increased in people with mild disease. IP-10, which is an important chemokine for recruiting T cells, PD-L1 is another sign of activation of T cells, members of the TNF family. But individuals with severe disease have a lot more higher levels of several more immune mediators, including what I'm sorry, my animations are not coming through. Oh, there they are. Okay, so I wanted to show these high levels of MCP1, which is a monocyte chemokine, a recruiting factor, as well as very significant increases in levels of platelet-derived growth factors. And this correlates really nicely with what we know in terms of thrombotic events that could occur in the context of severe COVID-19. So we definitely see signs of the cytokine storm here. Since monocytes are an important contributing cell type to cytokine storms, we decided to look at monocyte frequencies using flow cytometry. And you can see here that the number of CD14 positive monocytes in the peripheral blood does increase with infection. So we do have this monocytosis that occurs in individuals who end up having to be hospitalized. And this increase in total monocytes is driven primarily by CD16. So these are activated monocytes. But what was really shocking and surprising to us is when we looked at HLA-DR expression, which is an MHC class II molecule, very important for presenting antigens to T cells and getting T cell responses off the ground, the levels of HLA-DR was actually quite suppressed in monocytes from both individuals who are hospitalized as well as individuals who are asymptomatic or had mild disease. CD86 is also an important co-stimulatory molecule, so it's critical to engage T cells and maintain good contact between the monocytes and the T cells to get that T cell response off the ground. And those levels were also significantly decreased with disease. And so that prompted us to look a little bit more closely at other factors that we know are important for the antigen-presenting function of monocytes. And so we took another large set of samples, and then we looked for the expression again of HLA-DR, but other co-stimulatory molecules such as CD40, Fc gamma receptor, which is important for antibody-mediated endocytosis, as well as CD11b, an important integrin that allows monocytes to migrate. And we looked at the expression of these molecules relative to days post-infection, post-symptom onset. And what we see is that there is a very statistically significant downward slope in all of these factors with with infection and with progression of disease. And so the next question was, okay, so if these cells have lower expression of these costimulatory molecules, what does that mean in terms of their ability to respond to potentially a secondary infection? And so we purified monocytes, stimulated them with either viral agonists or bacterial agonists, and looked at their ability to make type 1 interferon. And what we saw is actually with days post symptoms, you have this steady increase in the amount of type 1 interferon that is made by these cells. Now, at first, you might think that that's a great thing. Of course, we want lots of type 1 interferon to be made in response to a viral infection or bacterial infection. But actually, there is such a thing as too much of a good thing. And increased levels of type 1 interferon that are not quickly controlled lead to dysfunction of the immune response, especially of the T cell response. So this is actually not a great thing. What we want to see is more of a bell-shaped curve where you have an increase in type 1 interferon that comes back down quite quickly. This is not a good sign. And then when we measure other NF-kappa B mediated cytokines such as IL-6 and TNF-alpha, which I'm not showing here, I'm just showing you IL-6 for an example, we actually see that these monocytes are incapable of making IL-6 in the case of viral agonists, or have a very reduced ability to make IL-6 in the context of bacterial agonists. So these monocytes have completely been rewired to make unhealthy levels of type 1 interferon, and they're unable to respond to a secondary bacterial or viral challenge, which could set up these patients for complications with a secondary infection. So staying on the theme of antigen presentation, after looking at monocytes, we decided to look at dendritic cells, because dendritic cells are really the primary antigen presenting cell for getting a T cell response off the ground. And in contrast to monocytes, what we saw was a profound depletion of dendritic cells in the peripheral blood, especially that of plasmacytoid dendritic cells, which are your primary type 1 interferon making cells and are potent antiviral subset of dendritic cells. Myeloid dendritic cells are also very important for antigen presentation, and those numbers were reduced. So this doesn't mean that they're dying. It doesn't mean that the virus is killing them. This could very easily be explained by the fact that these cells are being recruited into the lung, which is where they are most needed. Unfortunately, we were not able to obtain any BAL specimens. We would love to look at concurrent blood and BAL specimens during the course of infection. So maybe Dr. DeWitt and I will have another conversation about using the non-human primate model to address some of these questions. But as described for monocytes, the levels of HLA-DR and CD86 were profoundly suppressed in these dendritic cells. And so again, indicating potential problems with antigen presentation. So because of these changes, we next wanted to ask what was happening in terms of T cell proliferation and T cell responses. And we were quite surprised actually to see that, so the levels of total CD4 or CD8 T cells did not really change with infection, although there was some redistribution of cell types. So initially there was sort of this disappearance of central memory cells that came back later. And that comeback was accompanied with a rebound in the number of proliferating or Ki67 positive cells. And then very little proliferation in the CD8 subset was seen, mostly in the asymptomatics or at later stages of disease. So this is greater than day 15 post this symptom onset. And so to that kind of suggested that maybe there are problems with T cell responses. So just to give you a context, in most other viral infections that I've studied, I see T cell responses starting to peak at day 14 post-symptom onset. So this was really delayed. And so the next thing that we did was order an overlapping peptide library for the entire proteome of SARS-CoV-2. And then we pooled these into different groups based on their location in the genome and the size of the protein that these genes encoded. And so to give you a reference, these are in red are T cell responses as measured by the frequency of interferon gamma spot-forming cells in 200,000 PBMCs that we plate. So in any samples that are seronegative, we see no responses. Or very low responses. And I know this is controversial because there's been a number of papers that have said that there are a lot of cross-reactive T cells in circulation. Those studies actually report really small percentages if they actually look at interferon gamma, but for the most part, they actually look at activation markers by flow cytometry. We're looking at how many cells can make interferon gamma, so it's a slightly different question. But individuals who had mild disease and recovered, so most of these samples come from a healthcare worker surveillance study that we have ongoing that I don't have time to talk about today, as well as some people with very mild disease in acute stages. And in those individuals, we can see really robust interferon gamma production in response to peptide stimulation. But in purple, these are our hospitalized patients. And we have very few individuals who are actually making any discernible interferon gamma levels. So this goes hand in hand with the lack of that robust proliferation that we saw with flow cytometry. So we're going to use a different strategy, and we're actually going to use a high sensitivity interferon gamma ELISA to measure interferon gamma levels, because we hypothesize that there must be some antigen-specific cells, but using only 200,000 PBMCs for the ELISpot may not be giving us enough cells to detect those responses. And whereas measuring total interferon gamma in the supernatant may be a more sensitive way to do it. And indeed, if we do that, we can actually detect responses in both convalescent as well as acutely hospitalized patients. But I will draw your attention to this. So this is the amount of interferon gamma in the supernatant following CD3 stimulation. And in both the mild individuals who had mild disease as well as those who had severe disease, the levels of interferon gamma are significantly lower than those detected in seronegative healthy individuals who don't have SARS-CoV-2 infection. So there's definitely some dysregulation of T cell responses that is occurring. Okay, and so NK cells are really important innate cells that are recruited early on to the site of infection. And we were able to see, again, this depletion of KLRG1 positive NK cells in the peripheral blood. And again, we think that this is actually a sign of them being recruited into the lung where they are most needed. And at later stages of the disease, we see this big rebound in granzyme B positive NK cells. And so these are NK cells that are capable of killing virally infected cells. And so this is an indication that towards this later stage of diseases, we definitely see this engagement of NK cells. And this might be a compensatory mechanism for the fact that we don't have very robust T cell responses still out here, which is days greater than 15 post-symptom onset. Okay, so flow cytometry can only give us so much insight into what's happening. And so we decided to leverage genomics tools, and we did single cell RNA sequencing on a very large number of samples. So this was, again, another heroic effort by my graduate students. We had 28 total patients whose samples were subjected to single cell RNA-seq. We had an N = 4 per age group for each condition. And then in addition to that, for individuals who had severe disease, we included 3 different time points and 2 patients who succumbed to disease. And so eventually, when we actually get through most of this data, we are hopeful that we'll be able to tease out signatures of disease severity within age groups, so correcting for age, because we will have older individuals who are healthy, older individuals with mild disease, older individuals with severe disease who recovered, and older individuals with severe disease who unfortunately did not survive. But just so that you can see that we can identify the major subsets of circulating immune cells and PBMCs. So using the markers indicated here, down here, we are able to identify subsets of T cells, including naive and central memory and effector memory T cells, as well as regulatory T cells. We can identify monocytes, both the CD14, CD16 negative, as well as those that express high levels of CD16, dendritic cells, progenitors, B cells, plasmablasts, so this tiny, tiny, tiny population. So we may actually only rely on our single cell RNA-seq data to fully understand the changes in plasmablasts over time and with infection and with age. We were also able to identify platelets and erythroid progenitors, as well as CD34 progenitors in this study. And so this is a stacked bar graph showing you changes in the various subsets of immune cells between healthy individuals, individuals with mild disease and severe disease. And I understand that this is a ton of data. So I kind of tried to summarize some of the take-home messages of the, this is at this point a 30,000-foot view of the data, but we definitely see increased frequencies of erythroids and platelets, especially in aged individuals with severe disease. And you can see that down here. So this is the gray and the pukey pink color down here. And you can't pull out these subsets in healthy individuals at all, but you see a massive expansion of them with severe disease in both the young, but especially in the aged, where we see that erythroid compartment really become quite significant. And so this is again in line with the increased prevalence of thrombotic events in this age group. We also saw an increased frequency of NKT cells, which we did not look for by flow cytometry, but this is great so that we can detect this population of cells, as well as CD16 positive NK cells in young individuals with severe disease. We saw expansion of CD8 memory T cells in both young and aged individuals who have severe disease. And we definitely see an increase in the plasma blast cells with blood, and that increase was most significant in asymptomatic individuals and those with mild disease, in line with that higher level of antibodies at the onset. And then we also picked out a specific subset of proliferating T cells that will allow us hopefully to tease apart, maybe they were missed by our traditional flow cytometry approaches. Okay, so I'll really briefly go over this because I think I'm running out of time. But when we just compare severe to mild, so we're looking at disease severity and we're just comparing the transcriptome of monocytes in young or older individuals who have either severe or mild disease. The big thing that jumps out at us is individuals with severe disease. There is a massive upregulation of alarmins such as S100, A10, as well as other integrins that are important for monocyte migration. And you can see that also here in the aged individuals, a number of alarmins, as well as chemokines that are important for them to migrate, and a downregulation of some of the ISGs. And here is a downregulation of HLA-DR. So again, very consistent with our flow cytometry data. If we look at NK cells, we see that, again, severe disease is associated with an upregulation of genes that are important for dealing with either generating an inflammatory response, such as NF-kappa B, REL in the case of young individuals. And in the aged individuals, we also see this upregulation of genes such as CRAM, which are really important for inhibiting viral replication. So this may actually indicate that we may be able to detect some viral gene expression in these samples. So we will look at that. Fc gamma receptor gene was downregulated, again, in line with our flow cytometry data. And then finally, in the CD8 effector memory T cells, we actually see a number of interferon response genes that are upregulated with severe disease, both in the young and in the aged, and that might be in response to the high levels of type 1 interferon that are circulating. The other thing that is apparent in the older individuals is this increase in cytolytic molecules, CREM, granzyme B, as well as CD8 and other chemokines. Again, in line with fighting off probably a larger viral inoculum at this stage. Okay, so to summarize, we see that mild COVID-19 disease is associated with a robust but well-controlled inflammatory response, early production of antibodies, and higher frequency of plasma blast cells, whereas severe COVID-19 disease is associated with a cytokine storm that includes coagulation of coagulopathy markers, monocytosis, but loss of DCs in the periphery, immune suppression and reduced function for antigen presentation by both monocytes and dendritic cells, but increased frequency of cytolytic NK cells, potentially as a compensatory mechanism for the dampened T cell response in the lymphopenia that we see. Our single-cell RNA-seq data is already giving us some hints about novel age and disease severity-associated transcriptional changes. We are really looking forward to spending more time with this data and understanding the full ramifications of what it presents. And our next steps, we're really looking forward to actually look at the analyses, so single-cell RNA-seq analyses to understand the factors that modulate disease outcomes in young versus aged individuals, as I mentioned earlier. Because we actually have Asian individuals who are healthy and mild and with mild disease, and then severe disease with recovery and severe disease with death, we'll be able to really tease apart signatures that may be predictive of disease outcome. We're really interested in profiling the antigen-specific T and B cell responses to understand how repertoire changes inform disease severity. We would love to collaborate with someone where we can get access to BAL samples in order to address if the changes in the peripheral blood are in some way indicative of cell migration into the site of infection. And then we really want to know if there are long-term immunological consequences of severe COVID-19 disease. So with that, I will end. I owe my lab a huge, huge thank you. When everything shut down around the country and we got shelter-in-place orders, my lab really asked how we are a viral immunology lab. How can we, what can we do? And we all got to work. People worked shifts at all hours of the day, weekends, to go to UCI MC and obtain blood samples from the ICTS and process them, hundreds of antibody plates. So I owe them all a huge thank you. Thank you guys. And then of course, this would not have been possible if it wasn't for Frank and Dan, who really made it possible for us to get access to these discarded specimens from the remdesivir trials. Delia and Rob for tirelessly getting us discarded CBC samples from asymptomatic and patients who are not part of clinical trials. Dr. Alpesh Amin was instrumental in allowing us access to these samples. Dr. Chow provided us with a lot of metadata that we're still working through. Of course, the high throughput genomics core and the IFI flow cytometry core for sorting 28 samples in very short notice. And Melanie made sure that all these single cell RNA-seq libraries were done very quickly. This work was initially supported by a grant from the UCI Office of Research, and now it is supported by a supplement from NCATS to Dan Cooper, on which I am also a co-investigator. And I thank you all for your attention today, and I'll take any questions that you may have. Thank you.
Thank you very much, Dr. Messaoudi. Yes, we have a couple of questions that I want to get through quickly here. So the first is from Dr. Tom Lane. He's asking whether the expression of the T-cell chemoattractant chemokine CXCL9 and CXCL10 in the lungs correlate with worse disease or better outcome.
Okay, so we did not look at cytokines or chemokine levels in the lungs, unfortunately, because we did not have access to BALs, but we did look at them in the plasma. And we are running random forests right now to try and understand which one of those circulating factors is associated with disease outcome. Our initial preliminary analysis actually showed that PD-L1 and IP-10 are more indicative of worse disease outcome than any of those two chemokines.
Okay, thank you. And the next question is by Dr. Mike Bookmeyer. How would you summarize the response of the aged patient? Is the severity due to an immune senescence or an overreaction of the interferon response, or combination of these?
So all of the above. So what we think is happening is that there is it constrains on the T-cell response due to immunosenescence. And we know now with age that we accumulate these effector memory T-cells that are not really great at proliferating and are really specific for other antigens that we've encountered over life. And our naive T-cell compartment, which is actually what we need to respond to novel and emerging viruses, goes significantly down with age. And so I think to compensate for the lack or the dampened T-cell response, the innate immune branch goes into hyperdrive. And that's what leads to the cytokine storm, and that's what leads to these unfortunate outcomes. And I'm really looking forward to doing the analysis of the single cell RNA-seq data to really pull apart, dive deep into those particular subsets and compare the monocyte responses in individuals who are older and succumbed to disease versus those who either recovered or had mild disease. So what is unique about the older population who have mild disease?
Dr. Messaoudi, I wanted to extend on that question. And I'm sure you're familiar with these studies where they showed the development of these rogue antibodies against type 1 interferons in a subset of patients. So I was wondering what your thoughts were on this and whether that's something that you'll be looking at in your samples potentially. I believe these rogue antibodies against type 1 interferons, not the receptor, were found in about 14% of these really severe patients.
Yeah, no, I definitely read that paper with great interest. And in some levels, I wonder if the hyper production of type 1 interferon and the cascade of more and more and more and more interferon with time might be in response to the fact that their action is being blocked. Because we know that there isn't a whole lot of viremia in these patients. I don't know how the viral loads are in the lungs because we don't really look at that very carefully in the clinic right now. But yeah, that's very possible. And so yeah, we will definitely be looking at that. And I should mention that this cascade of higher levels of type 1 interferon is also seen in a lot of other viral hemorrhagic fevers such as Ebola. So that's a hallmark for us of like really severe disease outcomes in monkeys that are experimentally inoculated with Ebola, as well as patients who die from Ebola. So you see this cascade of interferon alpha that just keeps increasing over time.
Oh, very interesting. Yeah, the last question here from Dr. Perlman. He says from the single sequencing RNA data, you did not show neutrophils, which are the most abundant cell types in the blood. Can you comment on these?
I can definitely comment on that. So Eric, as you know, we were not able to get fresh samples. These are all discarded specimens. And so we did have to do a fecal purification and we only got PBMCs and neutrophils. Your beloved cells are highly sensitive to being outside for 48 hours before being processed. And so hopefully in the future, we'll be able to get prospective samples and be able to address those questions. Or maybe you will address those questions because I'm not a neutrophil biologist.
And then one last question that just squeezed in now, but have you looked at NK cytotoxic function other than just looking at markers or looking at those markers?
Yes, we have not. And we really, so we have to look at the limited number of PBMCs that we have left, but that would be a really interesting assessment is the increasing granzyme B positive NK cells really correlated with cytotoxic function, or is it just an increase in the expression of this one molecule?
Yes, wonderful. Thank you very much, Dr. Messaoudi. Very enlightening talk. With that, I would like to now announce that we will be going into a short break. After the break, we will resume with the following sessions 4 through 6, I believe, or I believe that's how many we have scheduled. So at this time, we're going to take a break and we will resume shortly.
Yeah, we'll reconvene at 10:45.
Thank you.
Great, your slides are coming through perfectly.
Thank you.
Anyone else need to check their audio?
Hi, it's Bernadette. Can you hear me?
Yes, we can.
Perfect.
Hello, it's Sunny. Audio check. Can you hear me?
Yes, we can. Let me enable a video, and then we can try sharing your video. Okay. Perfect. Video looks good.
Just a quick heads up that I'm Vladimir. Minin. I'm logging in from 2 devices so I can see what how the audience views the slides. Hope that's not too confusing.
Not at all.
Thank you.
And then Dino Bala, we saw your video, so you are good to go.
Thank you.
Should I do a video check? The last check says I cannot start the video. This is Sunny.
Oh yes, go ahead.
Still says, there it is. Does it work? Yeah,
Yes, it does.
thanks.
Thanks, everybody.
Hi, everyone. Welcome back to the second half of our symposium. My name is Sanghyuk Shin. I am an assistant professor at the Sue and Bill Gross School of Nursing, and also the director of the UCI Infectious Disease Science Initiative. So now I'd like to introduce our moderator for our session 4, Dr. Sunny Jiang, who is a professor and chair of civil and environmental engineering at UCI Samoli School of Engineering. Dr. Jiang, the floor is yours.
Great. Welcome back, everybody. Good morning. This is session 4, transmission modeling. It's my pleasure to introduce Professor Vladimir Minin, a professor of statistics at UC Irvine Donald Brand School of Information and Computer Science and associate director of the UCI Infectious Disease Science Initiative. After receiving PhD in biomathematics at UCLA, he spent 10 years on the faculty of the University of Washington before joining UCI in 2017. Dr. Minin's research revolves around developing rigorous solutions to problems that arise in biological sciences. The solutions often involve formulating stochastic models that can describe complex dynamics of biological systems and devising computational algorithm to fit those models to data. Dr. Minin is currently most active in infectious disease epidemiology, working on Bayesian estimation of disease transmission model parameters. His other interests include phylogenetics, population genetics, and system biology. Without further ado, please take it over, Vladimir.
Thank you so much, Dr. Jiang, for a kind introduction. I'm gonna share my screen now. All right, so thank you for having me in this very nice symposium. And today I will talk about our modeling efforts and analyzing local data from Orange County, California. So I want to start by saying that regional modeling of infectious disease dynamics is really hard, especially in real time. And to illustrate this, I'm showing you a tool from a graph from the CalCAT California COVID assessment tool that you can everyone can access and look at it. And this is the graph showing the RF, which is effective reproduction number. We will talk about in more detail what this means, but it's an important indicator. And you probably have seen it in popular press coverage of these numbers. It's an important indicator. We want this number to be below 1, which will indicate that we are controlling the outbreak or epidemic, or it's above 1, we're sort of in trouble because we have exponential growth of number of cases. And so this is the current snapshot of what California COVID assessment tool is reporting, and multiple teams right now reporting for Orange County these numbers, how these numbers changing over time. And the blue kind of solid thick blue line is the ensemble estimate that California Public Health Department sort of compiles and says, well, this is sort of some sort of average. It's not really average, but it's some summary of what multiple models are projecting. And as you can see, models are all over the place. So I think if UCLA thinks it's very low right now, RF for Orange County below 0.7. And then some of the, maybe Johns Hopkins, I think it's Johns Hopkins that says it's 1.2. So it's a wide disagreement. And that's not because these models are really inadequate, it's because it's a really hard estimation task. So that's what we will try to do in other things, but this is one of the motivations. And overall motivation is that there are many transmission models. And so, when we started working on that, we definitely asked ourselves a question, can we really contribute anything, or this is just going to be another model? And we thought there was one gap that we liked to, at least we attempted to fill, which was first using multiple streams of data at the same time to estimate the model. And we're not the only ones doing that, for sure. But also, we wanted to rigorously account for testing changes in testing over time, because we saw, and everyone has seen that, and everyone knows that number of tests performed in various parts of the country or even the world is changing. So the testing efficiency, or not efficiency, testing sort of capacity and number of tests performed varies a lot by space, across space and across time. And so what's lacking right now is these models explicitly accounting for that. So we hear a lot of, oh, cases went up because tests went up. Well, that's true, but instead of just waving our hands around it, can we just do something about it and say, okay, we have a model that accounts for that. So when we see increase in certain indicators, we already accounted for the increase of testing. So please don't point this out, because our model knows this and already took care of it. So that's what we're trying to do. One solution that is pretty common right now is dropping case data entirely and saying, okay, cases are unreliable because testing varies so much. And that's valid sort of mathematically and statistically. For example, looking only at hospitalization data is an okay approach. But all those indicators are lagging. Hospitalization goes off, chronologically comes after transmission events, after infections. So by looking at hospitalizations, one inevitably kind of looks at delayed data. And so that's also a problem. So ideally, we would like to use all the data, cases, deaths, hospitalizations, ICU beds, and all of that, and throw in genetic data into that as well. So ideally, it should be extreme data integration. We're not there yet, but I'll try to show you what we're up to. So the data we're going to work with is as I mentioned, this is regional data. So this is from Orange County, California. This is data provided to us by Orange County Health Care Agency, OCHCA. It is important that we actually we're not working with aggregators of, Johns Hopkins aggregators or New York Times county data. They're all okay data sets to look at broad picture of how transmission is changing over time and how it's different across different parts of the country. But after working with aggregated data and later with more cleaner version that OCHCA provides us, I came to the conclusion that actually these aggregators provide data that largely inadequate for modeling, really, and for statistical tasks, because it has lots of problems. For example, when we look at the data, we see a lot of repeat tests, because when you're admitted to the hospital, you will be tested multiple times, even if you're admitted for some procedure that has nothing to do with COVID-19. And those tests account for about, at least at some point in time, they accounted for 10% of all the tests reported by OCHCA. So not removing them exaggerates the number of tests performed and potentially skews your results. So that's a huge problem right there. Also, it's important to map the results of tests, positive and negative, to the actual time of tests, not at the times at which they were reported, which is another advantage of us using OCHCA data, because we know that when we construct our time series, we actually map the tests to these specimen sampling dates, not to the reporting dates. So we're going to first look at the retrospective data between March 31st and July 16th, and then we're going to look at the more current data using our situation report tool that we have online. So this is actually this is the full data set, the retrospective data. So this is tests performed, cases, which are just positive tests. So tests means positive and negative. Cases means just positive tests, and then deaths reported across time in the time period that I indicated. We bin those counts into 3-day periods, and that's a compromise. In epidemiology, it's custom to bin counts into weeks, right? Epi weeks. So we talk about weekly incidents. So in this case, this will be 3-day incidents. And this is a compromise for us because we want our if we bin into weeks, we have sort of less time points in a sense. And so by binning into 3 days, we are mitigating a little bit the weekend effect, so to speak, because we are, lumping Saturday and Sunday and Monday, or Sunday, Monday, and Tuesday, depending on where those 3 days land in the week. But at the same time, we're not looking at very, very noisy daily data, but we're not looking at too smooth weekly data. So it's a bit of a compromise, and allows us to do things a little better, we think. It's still, as you can see, it's still very noisy. It goes up and down, but it's important to realize that, I'm not too worried about these cases jumping too much, because tests jump with them. And so we're going to account for these features of the data when we take into account tests. The deaths being noisy is a little more problematic. So we don't have any control for it, so to speak. We don't have negative tests like we have for positive tests. So deaths are just inherently noisy because they're small counts. And so we do need to account for the fact that we observe only a small number of cases, and maybe larger, but still a fraction of deaths. So we have underreporting. And we also, of course, worry that cases may be preferentially sampled. So we don't assume that proportion of positive cases is really indicative of the proportion of transmissions in the population. So we want our generating model to account for that too. So our generating model is transmission model, surveillance model, and some priors. Transmission model is fairly standard SEIR type of model. We have 2 eye compartments just to indicate sort of early and progressed stage of the disease, and the fact that we probably catch individuals when they test a little later after they become infectious. And so we assume that when we talk about incidence, we're catching transition from this first eye compartment to the second eye compartment. Although we could include other transitions as well. And so we have, and then from the less infectious, from the progressed infectious compartment, an individual can transit either to the death compartment or to the recovered, removed compartment. So this is, and we model this using the model is simple, again, standard differential equation model that everyone is using under the sun right now, or at least the type of it. It's better if it's stochastic, but we are transitioning to the stochastic model right now. Right now we're using deterministic just for the, purely for computational efficiency, not because it's better. And the key parameters will be basic reproduction number and some mean latent period, mean early infectious period, mean progressed period. And so this is the relationship between them according to this model. And the important parameter, of course, is probability of death, or as it's known in epidemiology and right now also in the media, as infection fatality ratio. So this is not case fatality ratio, this is infection fatality ratio. So this is the probability that an infected individual will die. That's something we cannot really observe in the data. We have to impute it because we don't observe all the infections in the community. Our surveillance model has 2 parts: mortality surveillance model, which is very standard. That's something that people would always use in this setting. It's just some model that says, well, observed number of deaths is some noisy realization of the actual number of deaths. And we're probably undercounting because there's some individuals who die and never get tested, but probably the cause of death, if they were tested, would be attributed to COVID-19. So there's nothing particularly new in that part of the model. Our incidence surveillance model is new. It's not complicated, but it's fairly new, and because it accounts for testing. So basically, the key feature of that model that we don't confuse increase for testing for increase in COVID-19 incidence. And we allow for heterogeneity in this positivity ratio, which jumps quite a bit up and down because of testing guidelines are changing. And sometimes we're testing only symptomatic people. Sometimes we're relaxing that, depending on how are we rationing the tests? Do we have abundance of tests and we're not worried about it? Or maybe we're open campuses, as we do right now, and then the testing goes through the roof because we're testing students now a lot. So our model can absorb that these fluctuations into some parameter that controls how kind of jumpy that the test positivity is. Before we go into the results, which is probably the most interesting part, and that's why you are listening to me, not because you want to know more about statistical methodology, is that we need to talk about the interpretation a little bit. So basic and effective reproduction number. So basic reproduction number is this kind of spherical cow of infectious disease modeling, but it's a useful spherical cow in the sense that it tells us something about the speed with which transmissions occur. And so that number says how many people on average in one infectious individual would infect if it's dropped kind of artificially into completely susceptible population. And below 1 is good, as we mentioned, because that means that on average, one infectious individual is not really replacing themselves sufficiently to sustain the epidemic with one or more infectious individuals. If it's above 1, that means that you have exponential growth. In a stochastic model, when number of infections is small, it still can, the outbreak could go extinct and number of cases go to 0, but it's unlikely. So above 1, not above 1 usually means that there will be an explosion of cases. Effective reproduction number is basically kind of the same thing as the basic reproduction number, except it takes into account that as time goes on, we have some of the people get infected and hopefully get immunity, which we don't know for sure yet again, but that's what we hope. And therefore, maybe our susceptible population is shrinking, and therefore we should be multiplying this R naught by the proportion still susceptible. So RE is always a little smaller than R naught, and RE is this effective reproduction number that we saw on the second slide when I showed you these kind of problematic curves that don't tell us anything reliable about RE right now in Orange County from this California COVID assessment tool. Okay, so this is what to be on lookout. It's not the only thing we want to estimate. I will show you results of and we can estimate other useful parameters, but that's one of the more actionable ones. So what are we interested in? So we're interested in did transmission dynamics change in Orange County, and if yes, then how and when? So in modeling terms, that means that we want to know if basic and or effective reproduction numbers did they change over time, because they depend on multiple things. They depend on properties of the virus, but they also depend on the property of the people's behavior, really, like how we are coming into contact with each other. What other parameters may have changed over time? How many individuals we have who already had the infection? How prevalence is changing over time, and can we forecast using this model? So those are all these questions that we go over, but it's a kind of a preview of what we want to see from any model, really, not only our model. And so this is the kind of first results where we have these results for 3 time periods. So first, it's important to kind of orient yourself. We have one period, March 31st to May 5th, then May 6th to June 10th, and June 11th to June 16th. And so in the first plots, I'm showing you the basic reproductive number. So right here, this R naught, which, and this is our prior, which means that before seeing the data, that's what we sort of think R naught can be potentially be, in what interval it can be. And so this is a very liberal prior. We knew already that at that time we were under the stay-at-home order, for example, in March, but we still wanted to kind of give the model a chance to recover the actual number without telling it what it should be. So we are saying a priori, before seeing the data, it could be anywhere between sort of 0.2 and 2 point something, 2.5, maybe even more. And then this is the next little violin plot is the our uncertainty about estimation. This is our represents posterior distribution, or the how much uncertainty we have after seeing the data, what this number is, and it says that it's most likely below 1. We have some probability of it being above 1, but we think it's most likely it's below 1. in the period from between March 31st to May 5th, which makes sense because this was the strictest period in terms of mitigation measures. The stay-at-home order was in place and people stopped driving around as much and so on. And so this is, the next period is, it shows that it started to increase a little bit. And by a little bit, really here where we kind of do the, in terms of effective reproductive number, it's by a little bit, I mean by something like 17% or something, it increases in the second period, and in the third period, it increases even more and becomes sort of squarely above 1. And so that's kind of the story in 3 periods, as our speed at which coronavirus was transmitting in Orange County was going up with every period, which is not too surprising because the mitigation measures were relaxed at the same time. So you would expect that to happen. Proportion of susceptible individuals, as you can see, starts pretty much at 100% initially. Again, our model has the prior, but this is the posterior that we are putting on it. And so this is our estimate. But then it starts dropping because there are more infections in the community. It drops very insignificantly in the second period. It's still, 98, 99%. But it drops more in the third period because that's where the surge started in sort of June, July, which we also saw in the hospitalization numbers that we're not using here. So I'm using here only cases and deaths to estimate this proportion of susceptibles. So you can see that it's not a lot, but it's our uncertainties. It could be anywhere between sort of 9 to 2%, from 2 to 9% of individuals who already had the infection and potentially are immune. And so this effective reproductive number is basically a product of the first two. So this is R0 times this number will give you effective reproductive number for the first period. And because the proportion of susceptible is nearly 100%, the effective and basic reproductive number are nearly identical in the first period. So on May 5th, the effective reproductive number is the same as the basic reproductive number. But it starts going down a little bit and it changes because the number of proportion of susceptibles dropping with every period. And so in the last period on July 16th, for example, it's a little lower than a basic reproductive number, but still above 1. So it still looks like we had a exponential growth during that period. Again, this is just to kind of illustrate that by how much transmission increased, the speed of transmission increased by. So this is just taking the ratio of the REs in this consecutive periods and plotting the uncertainty around it. And this is the infection to fatality ratio. Again, this is our prior that we put on it, and so we think it should be below 1%, maybe with like some tiny probability above 2%, but that's kind of that goes into the crazy land. And then what we see in the posterior that it was dropping and with every period, and the third period dropped a lot. And again, even though our model is not taking into account age yet, which we should have an age structured SIR model, we don't have it at the moment. But most likely, as across all the country in the same period, this drop in IFR is mostly indicative of the fact that a younger population was being infected, and they have less probability of dying from COVID-19. So that's again expected and kind of comforting result that comforting in a sense that they intuitively make sense, and they agree with the results from other parts of the country. This is the part where we are kind of peeking into the unknown part, so to speak, where we're trying to get at what we cannot really ever see from the surveillance data. And so it shows cumulative deaths, cumulative incidents. So just one thing to notice, cumulative deaths are just a little bit above the observed deaths, which is what we want to see, in a sense that we think that most deaths are reported. We don't have a huge underreporting going on, but still we believe that they are underreported. compare that to the incidence, of course, we think that incidence should be hugely underreported, and that's what we see here as well. So the incidence, the latent incidence, the latent number of infections in the community is much, much larger than the observed one. To me, the most interesting part of this is this the last plot, which shows the prevalence. So that prevalence means at each time point, how many infectious individuals you have in the community on this day, at this time. And so as you can see in the first period, we were successfully suppressing the outbreak. And so our prevalence was going down. We never fully suppressed it, right? So as you can see, the number of infectious individuals in Orange County was, according to our estimates, was sort of in the range of 5,000–10,000. So it's not too small, but it was going down. But if you compare it to the next two periods, we went into the tens of thousands in the second period and potentially into hundreds of thousands by the end of the third period. Or it's not actually end of the third period. The last period here is our projection. So really the data that we used in this exercise at least stopped here, even though we have more data now. But just for the sake of argument, we kind of froze the data. And so the right-hand side of the plot, for example, in this plot that I'm highlighting here, is the projections. So maybe we were not in the 100,000s, but we could have been in the 100,000s by beginning of August in terms of number of infectious people walking in Orange County sort of on one particular day. So to me, that's the prevalence, these estimated prevalence is useful because you could start maybe thinking about risks in a slightly more precise language. You can say, well, if my prevalence is this, that means a certain percentage of Orange County is potentially infectious on each particular day. So if I open campus or school, I expect this percentage of people maybe to be infectious, and you can sort of do a back of the envelope calculation and see how many infectious individuals should you expect when you open a business or educational facility. This is our model diagnostics and forecasting. The forecasting is a tricky business here, so I'm actually showing it even though the results are not great in some periods because the mitigation measures are changing. So it's really hard to predict what would happen when stay-at-home order will be relaxed. So we are predicting test positivity, this is the bottom, pretty well in the first period, but deaths we are under. Our forecast underestimates the projected number of deaths because our model thinks that the prevalence will still be going down and the number of deaths will be declining, but it actually was not the case in this. And similarly, in this period, we're predicting, because deaths are lagging, we're still predicting them pretty well in the second period, but the positivity starts to go through the roof. After more businesses have opened in June, and so we're not predicting positivity anymore very well. So I just want to highlight that this prediction, when people are changing their behavior and government changes the guidelines, it's very, very tricky. It's not sometimes probably impossible to do it really well. I want to end with the today kind of the situation report that we updated yesterday. Yeah, but even though this is an online resource that we are updating every week with the help of Orange County Healthcare Agency data. Even though we updated it yesterday, notice that the report time period is from August 17 to September 21. And that's because we're still not sure that we can do very good job at dealing with reporting delays, especially with deaths. So deaths are underreported. So not they're underreported. That's okay. We can handle that. They are. The reporting of deaths are delayed by 2-3 weeks in many cases. And so the deaths reported today really most of the time means that death that happened 2 weeks ago, sometimes 3 weeks ago. So our model still gets a little bit confused sometimes when it sees that. The delays in testing results is not too bad because remember, we have a negative control in a sense. We have negative tests that we're using as well. So unless positive tests and negative tests are different in terms of by how much the results are delayed, which I think is unlikely, but we're still investigating how changing over time. then I think our model, our surveillance model for incidence is okay, can handle the delays, but deaths are problematic right now, so we need to downweight them. So that's why we are kind of being conservative, and our reporting period is, we're always stepping 10 days, sometimes more back to do it. So that's what we are producing. We again, this is our prevalence. Notice the prevalence we're projecting kind of into is still pretty high because we climbed pretty high in July. This is our prediction for the number of deaths, and this is our prediction for the positivity percent. This is percent of positive tests. And this is our current estimate of R0, which is below 1, but starts to touch 1. And this is our current estimate of infection to fatality ratio. And this is our historical estimates since we launched the report online. We have more historical estimates because we have been doing this reporting not online for longer, but this is online. We started our first situation report, went live with this June to July 25th period. And so that's how basic reproductive number has been tracking. It's been kind of bouncing around some value below 1.6, 0.7 or something. In the yesterday, it kind of jumped a little more. Again, maybe just normal fluctuation, or maybe we do see some increase. This one jump doesn't really mean yet anything, but it's the highest it's jumped since kind of June, July time periods. So it's a little bit not too worrying, I would say, but I think we need to keep an eye on it. Oh, that's bad. My collaborator disappeared. Oh no, here they are. So this is a work that has been done by lots of people, and I want to highlight first the joint authors on the paper we wrote about this, and graduate students Damon Bayer and Isaac Goldstein, who did a lot of work, and my former PhD student John Fincy, who is right now at biostatistician at NIH. So John has done a lot of initial modeling, and Damon and Isaac took it and ran with it. And then lots of collaborators, Daniel Parker and Bernadette Bodden-Albala from UC Irvine, lots of NIH collaborators who helped us along the way, and of course, Orange County Healthcare Agency, who has helped us a lot with the data. And support from acknowledging the support from the UC Irvine Council on Research, Computing, and Libraries. And so as you can see, this is my last slide. So you can get our situation report, which we update every week on Monday, Tuesday. The model implementation is available online. Everything is open source. You can look at what we've done, and we try to document it pretty well. And the manuscript is available on archive describing the methodology.
Great.
Thank you.
Thank you. Yeah, thank you, Vladimir. We have about a minute for questions. Let me read off the question box right now for you. I will start the video right now. Okay. Here are a few questions for you. First question asking, in your past research, you studied positive selection in various organisms. And to your knowledge, is there evidence of positive selection in SARS-CoV-2?
You know, it's a great question, and I probably can say something semi-intelligent about it, but I will. I think it's a better question for Dr. Andersen, who is going to speak after me. So, because he's going to talk about evolutionary aspects of the of this. So I don't want to steal Kristian's thunder.
Sounds great. Well, the second question talk about can you factor in control strategies to estimate their efficiency?
You know, we could, and when people have done it, we purposefully did not do this in this modeling, because in that case we would be sort of forcing the model to say that mitigation strategies do this or that. We prefer to do this sort of, so to speak, non-parametrically, and just estimate changes in R0, and then you could interpret that, okay, they changed because of the strategies, but it's the interpretation is very tricky. So I would like to be a little kind of open-minded about whether this was because mitigation strategies worked, or because people changed their behavior without really paying attention to what the mitigation strategies were. That's the hard question, but that's a good question, and we will try to kind of think more about it. But right now, we're not doing it. So.
Great, thank you. I got a reminder, this is a little bit over time. There are a few questions on the question box.
Sonny, you can go
Maybe
ahead.
you can address
Go ahead and
that.
deal with those, Sonny.
We deal with it? Okay.
Yeah,
sounds
you can deal with
great.
those.
Yeah, okay. Yeah, well, then I can ask the next question in the box. It said, can you break down the incidence of disease, hospitalization, and death by city in the Orange County?
Great question. We already are doing this. So we have a resource another resource, not the situation report, but data visualization resource that you could also access from the link that I provided, where we do this breakdown by cities, not all cities yet, but by, we started with 5 most populous cities in Orange County. We cannot do this with hospitalization, unfortunately. That data is not kind of decomposable into city data, but the rest of the data we can, and it's already there.
Right. All right. Let's move on to the next question. It asks, what portion of the test are serological?
So we're not using any serology. So all of these are PCR tests. Serology, when we will have results of the great serological study that our public health colleagues ran in Orange County, we are looking forward to incorporating that information. It will be super useful for us, but we're not using it right now at all.
Great, thank you. And I have one more question in the box. Is the reason why your ROs are mostly below 1 the fact that it is average ROs and the actual distribution is skewed? Is that the case?
Well, yeah, it's a little tricky. So R naught is an average itself, but that average has a distribution because it's a parameter and it has uncertainty, the estimation that has uncertainty. So we are not modeling super spreading events, and the model is actually deterministic right now. So there's no distribution of the number of people that individual infects. So I think it's purely the artifact of all the skewness that you see. It's kind of how the data, sometimes it's the conflict between death data, the mortality data, and the incidence data, which skews this number in one way or another. But I would not read too much into that skew because until we switch to a stochastic model, which is hopefully very soon.
Great, thank you. Thank you for your time in here. For the time's sake, let's move on to the session 5. This concludes the section 4. So now it's my pleasure to introduce the next moderator, which is for session 5 is Bernadette Bodin Alabala, Director and Founding Dean in Program in Public Health and Professor of Population Health and Disease Prevention and the Department of Epidemiology at UCI. Please take it over. Thank you.
Thank you very much, and welcome to session 5, epidemiology, I should say. And I'm really proud to welcome Dr. Micaela Martinez, who comes from my old haunting grounds, Columbia University. Dr. Martinez is an infectious disease ecologist. Her primary focus is understanding the drivers that shape seasonality in infectious disease systems, with particular interest in the impact of biological rhythms on disease. Her current projects aim to inform vaccination policy by revealing how demographic, physiological, environmental factors intersect at epi-prone disease systems, including polio, measles, chickenpox, and now, of course, COVID. Dr. Martinez conducts research on maternal immunity in infants and is building a statistical inference pipeline for studying vaccine modes of action. She utilizes cross-cutting statistical inference technologies and mathematical models to couple disease incidence data with clinical data to gain insight into the transmission dynamics of disease. And we are really excited to have her here talking today on, just finding your title, Micaela, social distancing, inequity, and racial disparities. Dr. Martinez.
Thank you so much for that introduction. And I will just take 1 second to share my screen, get my PowerPoint up. Here we go. Hopefully everyone can see that. So today I'm going to talk to you about racial disparities and social distancing inequities in COVID-19. I'm based at Columbia University, which is in New York City. And my lab, with the start of the pandemic and us being hit particularly hard here in New York, responded to the public health crisis and found that there was great need to address some of the racial disparities that have emerged during this pandemic. So I wanted to share with you one of our studies in this area. And so this work is in collaboration with colleagues at Boston University in the School of Public Health. And we have a paper that's going to be coming out in American Journal of Epi covering everything that I share here. But you can also find this paper on bioRxiv and my website. So just to give you a little bit of a background of the context of COVID-19 in New York, what I'm showing here in the large time series are the new daily confirmed cases in New York. And these cases are stratified by borough. So in New York City, we have 5 boroughs, and these are equivalent to counties in other locations. And so what I want you to focus your eye to is this beginning of the epidemic. So in New York City, as I said earlier, we were hit particularly hard in March and April. And the data that I'm using throughout the talk today is going to be focused on this March-April period when we were still in the upswing of the pandemic. And we were right about to start hitting the flattening of our curve. The inset here is showing you the cumulative cases. And the reason why I showed the inset here is that, again, I have this broken down by borough. And what I want you to notice here is that there are quite large differences in the cumulative cases per 100,000 people in each borough, with the Bronx being at the top. And so in New York City, even before we had COVID data released based on race and ethnicity, we had some signature or indication that communities of color were being hit particularly hard, and in particular Black and Latino communities, because New York City is a highly segregated city, and we know that the Bronx and Queens are boroughs that are predominantly Black and Latino, and these were also the boroughs that we were seeing having very high case counts at the beginning. And so one of the questions that we wanted to address was, we saw that there was variation from borough to borough in terms of the cases, also hospitalizations and deaths, which I'll show you some data later. But New York went into a very strong lockdown starting in March. We closed our schools. We had pretty strict stay-at-home orders. And one thing that's pretty unique with New York City compared to a lot of other places is that we can actually access mobility data quite well, quite easily, because so many New Yorkers rely on using the subway. So here's a picture on the top of New York subway being very, very packed pre-pandemic. This is kind of the typical New York subway. And then here's a more recent photo from the New York Times of how empty some of the subway lines can be. And so what we did was, we were interested in whether or not some of the variation we are seeing from borough to borough in cases had to do with mobility and the ability for people to be able to stay home, essentially, or flee the city. There had been news that in some of the most affluent neighborhoods in the city, up to 40% of residents had left New York and gone to homes outside the city. And so we wanted to dig into this a little bit more. What I'm showing you on the left are subway ridership data, which we were able to get for every single zip code. We actually got subway turnstile swipes for all of the hundreds of turnstiles within the city. And then we aggregated those into zip code tabulation areas, which are essentially small neighborhoods. And what I have is each of these color coded based on the borough that they're in. And what I want you to take away from this is, in the pre-pandemic phase, which was February and early March, which is on the left-hand side of the plot, we had the average subway ridership was, these are standardized. So average subway ridership pre-pandemic set at 0. And then as we went into the lockdown phase, we saw this very dramatic decline in subway ridership. but there's a lot of variation here. You can see some of these neighborhoods at the very top, especially in Queens that are in purple, didn't have as drastic as a decline as we were seeing in some of the Manhattan neighborhoods. And the averages by borough are shown in the thicker colored lines. And so we saw this variation in the essentially extent to which people stopped riding the subway from neighborhood to neighborhood. And we wanted to know, are there any predictors of which neighborhoods had a great decline in subway ridership and which did not? And so I wanted to show you these data spatially. So each of these little polygons is showing the 126 neighborhoods across the boroughs for which we had the subway data. And going from A through E, this is this decrease in subway ridership through time. So back in February, before our shutdown, there wasn't very much decline in subway ridership. There was a little bit due to people hearing about COVID cases in the news, but we had not established community transmission here yet. But then when you get down to panel C, March 14th, you start to see some of the lightening of these colors, indicating that some neighborhoods were starting to have quite a drastic decline in subway ridership. And then by April, we have this very homogeneous landscape in terms of how much of a decrease in subway ridership there was throughout the city. Now, since we have these 126 neighborhoods, and we also know a lot of details about these neighborhoods in terms of demographic composition, socioeconomics, and so on, we were able to ask the question, was there any relationship between how much a neighborhood declined in their subway ridership and mobility during the height of the pandemic and neighborhood demographics and socioeconomics? And one thing that came out quite starkly is if we actually binned subway ridership, binned the neighborhoods based on sextiles of median income. So we essentially took those 126 neighborhoods, broke them up into 6 categories based on the median income of those neighborhoods, and then plotted the subway ridership stratified by income. What we saw was a very dramatic relationship between income and the decline in subway ridership. If you follow the blue line, those are the most or the highest socioeconomic neighborhoods, the highest median incomes ranging from 62,000 to 147,000 median income, and they had this very stark decline in subway ridership, whereas the poorer neighborhoods were not afforded that same decline. And so what we wanted to do was statistically test this relationship. So we asked for these 126 neighborhoods, were there relationships between demographics such as the percent of their population over 75, median income, percent of people that were healthcare essential workers, percent uninsured, and also percent non-white. Was there a relationship between any of these socioeconomic categories and demographic categories and subway ridership? And I just need to pause for a second. Sorry. Living in New York, I have ambulances going by quite often. But what I want you to take away from this is if you look at this dashed line in black here, this is a line showing no effect of these parameters on subway ridership. And then those lines that are to the left or to the right is where we saw a significant effect. And so what we saw was that median income, the percent of the population that's in essential services, percent of population in healthcare services, and the percent of the population that is non-white, those were significant predictors for subway ridership during the height of the pandemic. And all of these, with the exception of median income, had a positive relationship, meaning that the higher percent of the population that was non-white, the more subway ridership you had during the height of the pandemic. And in terms of median income, the higher income related to lower subway ridership. And then we wanted to know whether or not this subway ridership and mobility during the height of the pandemic had any relationship whatsoever to COVID-19. So we have COVID-19 case data for each borough in the city, testing, hospitalizations, and deaths. And so what we wanted to do is to now factor this in to our model. And actually, we had even higher resolution data. We had the cases and testing and such, even on the neighborhood level. But I just wanted to show you these plots so that you could see what cases, testing, hospitalizations, and deaths look like. And one thing that I really would like to point out here is that each of these plots is stratified by borough. And if you look at cases and testing, you can see that Staten Island, which is our smallest borough, tends to be up here high on top under testing and cases. Now, Staten Island hasn't had much in the way of hospitalizations and deaths relative to the other boroughs. So what this has shown us is that there's some effect of testing on the cases that we observe. Staten Island has very high testing, and therefore they also rank quite high in terms of cases per 100,000, but you don't see that same thing propagate through when it comes to hospitalizations and deaths. So this was a signature to us that when we use case data, whether it be in transmission models or statistical models like I'm showing here, that we have to control for testing effort. And so what we did was we ran our statistical models to ask whether or not COVID-19 cases, when adjusted for testing effort, was related to subway ridership during the height of the pandemic, and then any of our socioeconomic and demographic variables. And again, what we found was that race, income, percent essential workers all influenced, now in this case, COVID-19 cases, and also subway ridership influenced COVID-19 cases. But the thing that was really important for us to recognize and see was that the subway ridership, race, essential work, income, all of these things were correlated with each other. And so median income was so highly correlated with the percent of the population that was non-white and all of these other variables that median income itself could capture a lot of the variation in COVID-19 cases. And so one of the other things that we are interested in addressing was, since we have this quite rich subway data in terms of seeing when different neighborhoods and when different boroughs started to have the rapid decline in subway use, we wanted to know if that had a direct influence on cases and if there was any temporal signature to this. What I want you to notice here or draw your eye to is, if you look at this red line, these are the subway data for Manhattan. And what you can see in Manhattan is that the decline in subway use, it starts to happen earlier than in some of the other boroughs. And then, of course, it declines more rapidly and falls to a lower level overall. But we wanted to know, did that earlier decline have any impact on COVID-19 cases? So what we did was we compared the subway data with the cumulative case data per 100,000 population in each borough. And in particular, what we did was a type of analysis called breakpoint regression. And what the breakpoint regression does, it essentially will identify when you have a transition point in some linear process. So we went from having the pre-pandemic subway use, which you can see here on the left at 0, and then we had these breakpoints which were indicating when the subway use started to decline. And each of these vertical lines here is showing when each borough started their decline in subway use. Now, we compared that to the COVID-19 cases at the same time, and what we call the flattening of the curve. So for each borough, we had the cumulative cases, which we were able to log transform and linearize, and then use this breakpoint regression to identify when was it that each borough stopped their exponential growth and had that beginning of the flattening of the curve. And what we found very consistently was that there was a 28-day lag between when we discovered that each borough had their decline in subway use and when they had the flattening of the curve for COVID-19. So what this shows quite directly is that you have these changes in the mobility that were influencing transmission in the community, and that there's this lag of 28 days, so nearly a month, between when you see that mobility intervention propagate in terms of slowing down of the cases. Now, what this has told us, kind of taken together, is that based on the data in New York, and I would argue that this would hold more generally across the United States, is that we have quite strong associations between race and ethnicity, where you live, how much money you make, and what type of work you do. And within communities, the percent of individuals who are essential workers. And this influences, in New York City, subway use, and in other cities, the correlate would be mobility in general. And all of these things are highly interconnected within the United States. And they come together to impact our ability to socially distance and to remain home when there are stay-at-home orders. And so overall, what we saw was that lower income neighborhoods in New York City had a higher burden of COVID-19. And these lower income neighborhoods also tend to be communities of color. But not only that, they are also carrying the burden of essential work for the city, and were the communities where were disproportionately having to go out to keep the city running. And when we saw within high subway use within communities during the height of the pandemic, we're assuming that individuals were continuing to use the subway during the height of the pandemic out of need, and it was out of need by necessity. And so it wasn't so much as a personal choice. And so this is why we term this social distancing and equity, in recognition that not all individuals have the same ability and essentially privilege to socially distance. And so social distancing isn't a nuisance, it's actually a privilege. And I just wanted to wrap up with a couple other comments, because this research has mostly been focused on mobility and transmission within communities, and that's why we focused on COVID-19 cases. Now, there's another side to this, which is that once an individual is infected with COVID-19, what's their chance of actually having severe disease, becoming hospitalized, or dying? And so here I'm showing the COVID-19 case data on the left, and then mortality data on the right, broken down by race and ethnicity for New York. And what I want you to notice is that consistently we have the purple and orange lines that are atop. And this is showing the disproportionate burden of COVID-19 cases and deaths within African Americans and Latinos. And so when we think about this, and the process that is happening is that you have individuals getting infected, and the types of variables that would influence your probability of getting infected include things like your number of close contacts, are you an essential worker, and the rate of infection in your local community. But contingent upon infection, those individuals that actually progress to severe disease, that can be influenced by pre-existing conditions, your access to healthcare, because that can influence how early you seek treatment and the treatment that you can get, and then also age, sex, and race. And so the way that I would like to propose that people should be framing this is we have this kind of two processes at play. The processes that impact your probability of getting infection, and then the processes that impact how severely diseased you get contingent upon infection. And I just wanted to leave you with a few facts I had put together. I put these together for our New York City Council, but I think that these are numbers that should be compared among other communities as well, and really just trying to highlight this very strong inequality that is existing with COVID-19. So Black and Hispanic New Yorkers are 2 times more likely to die of COVID-19 than their white counterparts. The Bronx and Queens have had 1.8 and 1.7 times higher COVID-19 death rates compared to Manhattan. And Manhattan, as I had mentioned before, is the highest income borough. Hypertension, diabetes, and obesity are 2.4 to 8 times more prevalent in the Bronx compared to our most affluent area, which is the financial district, Greenwich Village, and Soho. And so these fact 3, this incredibly high difference in terms of the prevalence of these pre-existing conditions can be one of the driving factors for why we see such exacerbated mortality rates in the Bronx and Queens and among the Black and Latino community. And then I just wanted to highlight that as of today, the top 3 comorbidities that we're seeing in New York for COVID-19 mortality are hypertension, diabetes, and hyperlipidemia. And these are diseases of our environment and lifestyle. And there has been much work in the environmental justice and social health justice literature looking at things like food deserts, access to green spaces, and how these things can influence these chronic health conditions that are now putting people at higher risk of COVID-19. And with that, I think I'm probably right about at time.
That was really terrific. Thank you very, very much. Let's go through some questions. Okay, let's try this one. So were you able to estimate how much of the race ethnic differences are explained by differences in subway use?
Were we able to? Yeah, well, yes and no. So what we could do is we can essentially have these correlations with the neighborhood composition and subway use. But the reason it's particularly difficult to tease apart, let's say, the contribution of race in addition to income or essential work is that those things are so highly connected with each other. And so and this is why I think expanding outside of New York would be great, where we can start teasing apart these factors a little bit more, or perhaps having studies where we go target particular groups where, let's say, perhaps you can control for income, but income and the type of work that you do, but then you have individuals that only differ in race and ethnicity to see if we could have any impacts there. But in terms of other factors that might be associated with race that would potentially could drive subway use variation that we didn't control for could be cultural things, because we did control for essential work, but also things like individuals living in multi-generational households. Maybe you have to go out to get prescription medicine and things, because this was at the very height of the pandemic, when if you weren't an essential worker, you were only able to go out for food or for medicine.
Right. And I would just say, so we've been studying this here in Orange County, where we have predominantly Latinx population, and finding very similar things, but we don't have a subway. And so I think, to your point, to be able to parse out, I think some folks are interested again in, subways themselves pose risk of exposure, especially touch, and of course air droplets, especially New York subways, if you don't have masks. I don't know if there's anything else you wanted to comment about that particular point.
Yes, so with this study in particular, we're using the subway use as a proxy for mobility and essentially people moving around their neighborhoods during the height of the pandemic. So we weren't able to address whether or not being on the subway itself, like riding the train in physically, if that itself was contributing to higher transmission, or just simply more people that are riding the subway, just the more contacts they're having on a daily basis, the more they're out.
And to what extent is that, again, I want to talk about population density versus race ethnicity?
Yeah, that's a great question. And this goes back to, especially when you're trying to tease apart race, ethnicity, and income,
Right.
because within New York, it's a very expensive place to live. And so when we essentially look at lower income households, then that's when, out of necessity, you may have to pack more people into, a relatively small apartment. And then because of income, and then also just culture, you can have multi-generational households. So you may have, 5 people in a household, 10 people in a household that are of three generations or even more.
Exactly. And then ridership frequencies. Here's an interesting question. How much ridership frequency between low-income areas and high-income areas are influenced by what we heard here on the West Coast, rumors of high-income folks vacating their homes during the pandemic?
Yes, so we absolutely had some of that especially in lower Manhattan. People were leaving out to homes in the Hamptons and upstate and such. But so we don't have the data on, if you swipe at one turnstile and let's say you're from the Bronx and you're an essential worker, did you go into lower Manhattan to work, or were you working in the Bronx? Right. We don't have the back and forth in terms of where people were going, but we can see that some neighborhoods have more essential workers that live in those neighborhoods. And grocery stores and hospitals and such were having to be run all over the city. And so the fact that you have essential workers are kind of geographically localized in terms of where they live, yet you have an entire city open, we know that some of these essential workers were having to go from, the boroughs like the Bronx and Queens to service boroughs like Manhattan. So we don't know the exact to and from where people live versus where they work and what the commute was, but there's some kind of basic inference we can draw.
And then a couple of questions about some social determinants of health, like testing and healthcare access. Do we know to what extent these are affecting the disparities in cases, and then in particular in deaths? And then just following up on that, have we controlled for in New York City, looking at disparities in death rates, have we controlled for some of these comorbidities?
Yeah, so to date, my lab has been collecting or collating the hospitalization and death data and working with Mount Sinai Hospital and Columbia Presbyterian, where I'm at, to try to get at some of the actual pathology, and we want to do that on an individual level with the hospital records. I don't think that these population level data are sufficient for that. But in terms of, I can't remember what I was going to say, I lost my train of thought. But in terms of this study, we were strictly focused on the acquisition of infection part of it. So we didn't, in the statistical analyses, I was not including hospitalization or mortality, because we were, with the subway data, all we can really address is the transmission side and not the pathology side. But that's the next thing that we would like to do, is really to get at some of the chronic diseases and seeing how they're really impacting risk.
That's great. So we in the social determinants of health field have long been advocating for policies, health policies, that do not actually open the gap, but in fact narrow the gap in terms of disparities like you've presented today in virus acquisition and in poor prognostic outcomes. So what would you say that an ideal policy would include if we were including a policy on social distancing, but that we really wanted to, we wanted to sort of equalize and narrow the disparities gap?
Yeah, I think that to narrow that, we really have to reallocate our efforts. For instance, here, when we were seeing the Bronx and Queens being hit so hard, yet in Manhattan was where we were having all of these temporary hospitals set up. And my colleagues and I argued or presented like why are we not setting up Yankee Stadium, which is in the South Bronx, to be able to have these temporary facilities? So with the allocation of resources is a big thing. And it doesn't, right now what we're seeing is that based on the testing efforts, as I had mentioned earlier, Staten Island has very high rates of testing. They do not have equivalent death and hospitalization rates as some of the other boroughs. So that leads you to ask, how are we actually putting resources out there? We shouldn't just say we need a certain number of tests per 100,000 when you know that some populations, they're going to have a much higher severe burden of disease. And so does that mean that they need to have 2 times as much resources for testing? Does that mean that we need to reallocate ventilators differently? And so I think that equity is not just standardizing things to population size, but really needing to go above and beyond to address these inequalities.
Thank you so much. This was really a very interesting and important talk. And I know anybody else who has any questions can email Dr. Martinez. I think we're moving on to our next session, which is session 6. And so thank you again, Dr. Martinez. And I want to introduce Dr. Shriti Gohil, who is an infectious disease clinician and epidemiologist, and will moderate this session. She's actually worked tremendously in the UCI efforts trying to reduce transmission of COVID, and Dr. Gohil is a researcher and an ID researcher in her own right. Thank you so much, Dr. Gohil.
Shruti, we can't hear you.
Shruti, we still can't hear you.
I can see audio isn't connected on her, either the laptop or iPhone.
We'll fix that, Kristian, don't worry. I hope anyway.
What are the last 2 digits of your number? Type it into the chat. Okay, try it now.
Okay, great. Thank you. It looks like I'm back on. Is that right? Can you hear me?
Yes.
Yeah, we can hear you.
Okay, great. Sorry for that. It's my great pleasure to be here. Thank you, Dr. Bowden. And I'm here to introduce Dr. Kristian Andersen. Absolutely a pleasure. Thank you for joining us today on this consortium. Kristian Andersen is a professor at Scripps Research and vice president of the Viral Hemorrhagic Fever Consortium, and has over the past decade spearheaded large international collaborations investigating the emergence and spread, as well as the evolution of viral pathogens like SARS-CoV-2, Zika, Ebola, West Nile, and Lassa virus. Dr. Kristian earned his doctoral degree at the University of Cambridge and performed his postdoctoral work in Paris Abedi's group at Harvard University, and at the Broad Institute, 2 giants in the field of genomics. So our great pleasure to have you here. And I will say one thing from a clinician standpoint and epidemiologist standpoint, that your work will be listening to very, very carefully, because that is all the talk of our world as the fall approaches, and a renewed concern over the possibility of a new strain or reinfection in our patients as flu comes in, and as we know the PCR technology to be persistently positive in patients who have old disease. So absolutely critical. Thank you, Dr. Andersen, for joining us.
Yeah, thank you, and thanks for having me. So today I'm going to talk, well, my title of my talk was to keep a genomic eye on this new virus, SARS-CoV-2. And we actually do a lot of different things when it comes to utilizing genomics. We try to understand spread of viruses, but also understanding some insights into these viruses themselves, what makes them tick, what makes them so special. And I'm going to spend a little bit of time talking about all those different things today. I'm actually going to take you back to the very beginning of this outbreak, and then going to take you up to what is happening right now, and also showing you some of the utility of genomics and how we can use that. And just to give a brief overview on the first part. So when we're talking about genomics, there's actually a lot of different reasons why we want to do that in the first place. It's really mentioned around emergence and evolution of these. We are definitely interested in those kinds of questions, and I'm going to show you some of that. But when we go to sort of the more applied aspects of using genomics, we're trying to get a better sense of epidemics, outbreaks, and pandemics from a genomics perspective. And what's really helpful here is that what we can do is that we have infected patients coming in, for example, this example here for a couple of different outbreaks. We sample some of these that are going to be a representation of the community transmission of this virus. And then we're going to directly sequence the virus from these individuals. And we're then going to use that information, and Vladimir had talked about that earlier today, going to use that information to create these phylogenetic trees, or just think of them as family trees, which essentially becomes a sampling of the transmission of that virus itself. And once we have that information, there's many different kinds of questions that we can now gain insights to, which you wouldn't be able to gain insights to if you did not have the genomic data itself. Things like how many introductions do you have into an area? And I'm going to show you some of those examples from the very beginning. Where did the virus come from? And there's multiple different questions on that. For example, where did it come from before it was in California? Where did it come from before it started the pandemic? Then as the outbreak goes on, we can start to understand aspects of local transmission. How is the local transmission sustained? Is it because it's locally spreading in the area, or is it because it's continuously being reintroduced, for example? As the outbreaks go down and start coming up again, which we're certainly starting to see many places in the United States as well as Europe now, trying to get an understanding of what drives these reintroductions, or what drives these flare-ups. Is it the virus that's been in the community all the time? Maybe it's been undetected because we're not looking, or is it actually that it's being introduced from the outside? But let me take you back to the very beginning of this pandemic and essentially get an understanding of what is going on. Trying to understand the origin, the very simple questions of where did this virus come from. What we are observing with SARS-CoV-2 and COVID-19 is, of course, something that's highly unusual. Pandemics of coronaviruses we have never seen before, and typically pandemics is something we see from the flu viruses. But now we have a new virus, and we were trying to understand really early on where did this virus actually come from and what's driving this. So going back to the very beginning of this, going back to where did this all get started? As you all remember, Wuhan was the first place where the virus was detected in Hubei province of China, pretty close to Guangdong, where we saw SARS-CoV emerging back in 2002 and causing an epidemic 2002, 2003. Wuhan was the place where SARS-CoV-2 was detected. And I'm saying detected because detection does not necessarily mean that that is actually where it came from. In the very beginning, we thought that that's where it's coming from, but I'm going to talk a little bit about how Wuhan doesn't necessarily have to be the ground zero of this particular virus, but is where we detected it in the first place. So when we saw this coming up and people started releasing genomic sequences, the first one was back in January 10th. Ilhem did that on behalf of his Chinese colleagues. And then over the following weeks, we got several different genomic sequences that were publicly released. And that drove us to a question which just had to do with when did all of this get started? We all know about the timing as reported by Chinese authorities. We know all when this came on our radar. And if we look back at that, it's sort of in the mid to end of December is when they start getting a realization of a novel outbreak. And then the world knows about this about a week and a half after that. But our question was different. It was essentially saying, well, but let's take the genomic data and let's ask ourselves the question as well as one thing is when you detect this thing, but when did this all actually get started? We can answer that based on genomics. So that's the first question we tried to address here. So again, just to take you back to Wuhan and all of this got started and it got on our radar was this Huanan market with the timing of December of 2019. This is when there was some unknown pneumonias that popped up that sort of alerted the system to there's something going on here. It's not flu, it's in the middle of flu season, right? So you have a lot of pneumonias in general, but they pretty quickly realized that actually this is not actually flu, it's something novel. It's something we don't know what it is. And all of this was originally linked to this seafood market, the Huanan market in Wuhan, again, mid to late December of 2019. Now, it has to be understood that back then, because of what we knew about SARS and MERS related coronaviruses, they don't really spread very well human to human. And all these initial cases were all linked to the seafood market. The idea was that the seafood market is probably the source of this particular outbreak. But it needs to be clear that we now know that SARS-CoV-2 is indeed spreading very efficiently human to human, right? So this seafood market is not necessarily the source here. It's very likely that the seafood market could simply have been a super spreading event where we detected the virus in the first place, but it's not necessarily where it got started. But once we had that, we go back and saying, okay, we're now in December here, but looking at these genomes, what do we actually find in terms of the timing of what was back then an outbreak before it became a pandemic? And when we look at that, what we see is that, well, once we have these viral genomes, we can sort of look at the evolutionary rate of this virus, because viruses, every time they transmit, they mutate a little bit, on average, 2 mutations per month. And they come up at a very clock-like figure. It's essentially the same that we are all aging, some of us a little faster than others, especially during COVID. But viruses do the same thing, and they do that via acquiring mutations in the genome. And we can do that and see this evolutionary rate. So once we had a good sampling of the viral population, we can get this evolutionary rate, essentially the molecular clock of the virus. Once we had that, we can take that and saying, just start counting mutations in this family tree, phylogenetic tree that we have, and just ask ourselves the question, what is the time to the most recent common ancestor of all the viruses that we see circulating? And there's a few different ways of doing that. Let me see, that's not actually showing up. That's weird. Okay, let me just quickly move my screen share here. So on the right side of my screen, you can see that particular slide that won't actually show up. But what that shows is that if we do this in a couple of different ways of estimating that, what we find is that this timing of when this actually all got started just based on the genomics is something mid-November, early December. So very, very close to the time at which this was actually detected in the first place. Now, there's been a lot of talk about the delays, and China delayed telling the world about it and things like that. But when we think about it, we look at it from this aspect, it's actually only a couple of weeks from when we know all of this got started from a single source into the human population, detecting first that there's something unusual going on, and then only a week or two after that, fully sequencing the virus and knowing that this is a novel coronavirus and this is exactly what it looks like. That is exceptionally fast. If we look at detections, prior detections of even novel known pathogens like Ebola in West Africa, for example, there was a delay of 3 months from having the outbreak started to us realizing that actually these weird cases, they're Ebola cases, right? The reason being we didn't expect to find Ebola. in West Africa. We certainly should, but we didn't expect to find it, so we wasn't looking for it. This is a known pathogen, right? We already knew about Ebola, yet it took us 3 months to actually detect it. Look at Zika in the Americas, it's more than a year between that virus starting an outbreak in Brazil to us realizing actually a year and a half later that there's these mysterious cases of microcephaly and they're linked to Zika infection. So typically these timing events are much, much longer, and you have a really large gap between something starting and you actually realizing that. So this particular timing is really rather quick, to be perfectly honest. Now, once we had that data, the next thing we could look at is that how many times did this actually get into the human population? We believed in the beginning, again, for the Wuhan seafood market, maybe there's a lot of animals in there that are infected, and that's then what's infecting individuals. But in the following weeks, because of the release of genomic sequence from Chinese investigators, we could see that this is actually just a single event. And the reason why we can say that is that initially we had such limited diversity in our viral population. The virus was basically the same, whether it was individual 1 or individual 2 that showed up with infection. So what that shows us is that it is actually a single event. It doesn't mean that it's necessarily a single animal into a single human being, but it has to be a very small cluster of infected animals, recently infected animals, into a very small cluster of humans. And that's all what got that started. And past that point, it's all sustained human-to-human transmission. Now, this was really important early on because, again, let's remember that the belief was that there was limited human-to-human transmission because that's what we have learned from SARS and MERS, so that the same expectation was true here. But that's just not what we saw. It was actually very, very early on, all of this was driven by human-to-human transmission. And I would say that the genomic data certainly showed it a lot earlier than this realization from epidemiological data that this is human-to-human. There was speculation about it early on, but it wasn't really believed because the data was missing. The genomic data went in and actually added that. Now it's all obvious. Again, there's a lot of talk about the hiding that they were spreading human to human early on. That's not what it is, right? It's just, it takes time before we can actually catch up with the data. And again, the genomic data was some of the earliest here. Now, when we go back to this, some of the things that we were really sort of confused about was that what is going on here? Because typically, and I mentioned this early on, is that if you just look at the epidemiology really early on, which is some of the articles that I'm showing here, which is spreading through cruise ships really fast, spreading through hospital systems really fast, is not typically something that we actually see for non-flu viruses. It's really unusual. Zoonotic viruses in general, they hop into humans, typically pretty crappy, and it takes a while before they get going. But we saw with SARS-CoV-2 that this was really happening really early on. So we asked the question saying that why is that? What is it that makes this virus so special compared to all the other viruses that we have seen before it, including things like SARS? So just to show this is that the really unique feature about SARS-CoV-2, both compared to the flu viruses as well as compared to the coronaviruses and other viruses, is that it spreads really easily and it causes really severe disease. If you look at the flu viruses, you have something like seasonal flu, which spreads quite easily, right? But it doesn't really cause very severe disease. And the reason for that, again, sorry, my slides are messed up here for some reason. The reason for that is that seasonal flu stays up in the upper areas primarily, and then it stays there. If you look at the bird flu viruses, which obviously cause very severe disease in humans, but they don't spread very easily, it's because it gets deep down into the lungs. It stays into the lungs, right, and causes severe disease, but then it doesn't spread really easily because it's not high up. That is obviously all determined based on sialic acids and the receptors that these viruses use. But what is interesting is that when you look at SARS-CoV-2, it does both, right? It infects upper airways, infects lower airways, hence can spread really easily and cause pneumonias and severe disease. So if we go back and saying like, but what is that? Why is that? And why is that SARS-CoV-2 is so special here? So that turns us back to the genomic data and just ask a really simple question. Why? Why do we see this? And the first thing we say is like, well, where's this thing coming from in the first place? Right? We knew it was in something else before it came into humans. And the question is, where is that? Now, if we just look at the phylogenies of these viruses, really related viruses, the slide here is showing all the different SARS-CoV-2 sequences here and related viruses. And what we find is that these viruses are all circulating in bats. You have all these SARS-like coronaviruses that quite clearly have a bat reservoir. SARS-CoV-2 is no different here. Very likely a bat is the ultimate reservoir. But what is important though is that while there might have been an intermediate host, so between the bat and the humans, it's plausible that some sort of host existed where maybe the virus got a chance to evolve there a little bit, and then from there jumping into humans. We don't know what that host is. Pangolins have certainly been suggested many times. I don't think we have any data to suggest there really are, but it is a possibility. But something could have existed between the bats and the humans. So then we go, it's like, okay, now we know that. Let's now look at the features of the SARS-CoV-2 genome and see, can we learn anything about what makes this unique? What is it actually that makes this such a special virus? And when we did that, there was two things that really stood out to us. When we looked at SARS-CoV-2 and compared that to other coronaviruses and, SARS-like coronaviruses and SARS itself very specifically, is that we found two features which were quite unusual. The first thing was that the receptor binding domain of the virus, the part that actually binds to the human receptor, is the ACE2, the receptor, and that's mediated by the receptor binding domain, and there's 6 amino acids that determines this binding into the ACE2 receptor. What we found is that that receptor, and this was based on just computational studies, I should say, it's now been confirmed with experiments, but based on the computational studies and molecular evolution, is that it really looked like the binding between the SARS-2 receptor binding domain and the human version of ACE2 is a really strong binding, much stronger than SARS, for example, was when that jumped into human. And that was unusual. That was really very peculiar to me, because that should not really what you would expect, right? That would have evolved to a bat host or some other host, right? And now we saw that it bound really well to the human, specifically to the human version of the ACE2 receptor. We also found a polybasic cleavage site. So this is a little site that sits here, which was unique to SARS-CoV-2, not something we have seen in other SARS-like coronaviruses. And the reason why that's important is that this spike protein, which is the protein that binds to the receptor, it's on the outside of the virus, has to be cleaved between its two subunits, S1 and S2. And typically, it doesn't do this very well. And if it's not properly cleaved, while it can bind to the receptor, it can't actually infect the cell. SARS-CoV-2 had this neat little trick where it had this polybasic cleavage site, where it actually allows it to be processed really, really efficiently, and therefore being able to not only bind to cells really well, but also infect a large number of cells. So we think a lot of this was determined based on this polybasic cleavage site. And importantly, this cleavage is mediated by furin and other host enzymes, things that we have in our cells. So these were the primary things that made this such a unique virus. And this is just to show the features of like this high affinity binding, which again is something we looked at the structural modeling here, and we could really show that this was highly efficient for SARS-CoV-2 specifically. Now, the furin cleavage site itself is actually really interesting, because we know these, first of all, a lot of coronaviruses have these too. The common cold coronaviruses, half of those have furin cleavage site or polybasic cleavage sites. The SARS-like coronaviruses, we hadn't seen these before, but again, we see them in other coronaviruses, and really importantly in the flu viruses, this is sort of a hallmark feature of highly pathogenic avian influenza viruses, is that they obtain these furin cleavage sites, typically when they spread through really dense populations. And now we found the same thing in SARS-CoV-2. And we think that that furin cleavage site, together with this high affinity binding, is really what explains why this is such an efficient human pathogen. It allows them to infect many different organs, right? Not just binding to the receptor, and it allows it to bind really, really well to the receptor. So we hypothesized all of this back when we wrote our first paper on this, and since we have had experiments that actually showed that yes, this is true, we do know that these features play a really critical role in the virology of this particular virus. So once we answered that question, saying like, okay, so now we know why we had some ideas of why this is a human efficient pathogen, is that how did it obtain this? And of course, there's been a lot of talk, and there still is to this day, around this came from the lab, this came from bat, this came from whatever, right? And we wanted to look at all those possibilities, because I would say all of them are plausible explanations what happened here. Of course, our default is the natural evolution of the virus, because we've seen that occur so many other times. And what was interesting is, as we were going through this, what we actually found is that, well, this really high affinity receptor binding domain of SARS-CoV-2, we actually find exactly the same in pangolin viruses. That was something we didn't know when we saw the first genomes, because those pangolin viruses had not been sequenced yet. But during the first couple of months here, people found SARS-like coronaviruses that had exactly the same receptor binding domain. So you can see it here, but here's the really closely related bat. It has different residues in these binding domains, but in the pangolin virus specifically, it's actually exactly the same, right? There's another pangolin virus down here, and some of the bat SARS-likes pick up some of these residues. So this tells us like, well, wait a minute, this receptor binding domain is actually something that exists out there in nature. We already know that it's out there. We hadn't seen it before, but now we do. And specifically, we have seen it in these pangolin viruses. So taking that, other investigators looked at this a little bit more closely and saying like, how did that particular receptor binding domain actually happen to end up in SARS-CoV-2? And we don't fully know, but what we probably see is that in some historical event tens of years ago, is that there was some bat coronavirus that contained exactly that receptor binding domain shared between SARS-CoV-2 and the pangolin viruses. And then some of the more closely related bat coronaviruses like RITG13 lost them over evolutionary time, but they were retained in SARS-CoV-2 as well as in the pangolin viruses. And what's really interesting about these receptor binding domains in general is that there's this very recent paper, it actually came out last week from Shin from Wuhan Institute of Virology, is that there's this evolutionary arm race going on between ACE2, the receptor of these coronaviruses, and then this receptor binding domain of the virus. And specifically, this evolutionary arms race is going on in bats. And that's pretty scary, to be perfectly honest, because it means that these viruses are shuffling residues that bind to these ACE2 receptor all the time in its natural host, the bats. So the fact that we find one that binds really well to human just by chance is actually what we would just expect. It also means that this is something we should certainly expect to happen in future as well. The other thing that we found is that this furin cleavage site is that we haven't yet seen a full version of exactly the same in other SARS-like coronaviruses, but we have started to see evidence of other bat coronaviruses having insertions in exactly this region of the genome. So we know that it must exist out there yet. It's just we haven't observed it yet, because of course our ability to sequence viruses, of which there are very many, in bats, of which there are also very many, right, is extremely limited. But we're starting to see some remnants of exactly this evolutionary process going on. The other thing, again, speculation about this having been cultured in the lab, what's actually pretty interesting is that if you do culture SARS-CoV-2 in the lab, it actually loses that furin cleavage site. It's not something that's beneficial there. So this sort of rules out like the lab and shows this probably did not come from the lab, right? Which is important, but we sort of expected that. But again, we didn't know. And shows you that this is actually just a natural evolutionary process of coronaviruses happening all the time. And indeed, if you look at really early studies looking at exposures to SARS-like coronaviruses, many places of China, like Yunnan province, for example, we know that this has happened, something that happens frequently. Importantly, though, not really something that we have seen evidence of around Wuhan. They have different bat species there. So we don't, and this is one of the reasons why I don't actually think that this necessarily got started in Wuhan. So going back to my previous question, which is that what then got this started? What is it actually that made this whole thing going? And we have two different hypotheses that we are hypothesizing here. One is the market hypothesis. It's probably not what we should call it, but what that is that you have a reservoir and the virus is basically fully formed there. And then either it jumps directly from the reservoir into humans and causes the outbreak leading to the pandemic, or it goes from that reservoir into an intermediate host that is still unknown. And then from there actually leads into humans and then it explodes there. The other hypothesis that we are entertaining is one that we call a pre-circulation hypothesis. And it comes to when did this virus acquire this furin cleavage site? We think it is possible that there could have been some ongoing pre-circulation of the virus that probably did not have this furin cleavage site, probably didn't really lead to disease. It wouldn't have been detected and also would have had a limited number of people that have been infected and therefore not really getting on the radar of public health authorities. So it could have done that for a few months, maybe as part of that process, gain that furin cleavage site. Again, the common cold coronaviruses have these, so we know it's good in humans, right? And then from there, take off into the outbreak and then into the pandemic. So we can't distinguish the two because if you just look at the data, they essentially look identical. So going from that and saying like, okay, we learned something about the pandemic and all of this, how did it actually get started? And our genomes allowed us to get some insights on that. But what about now? And now can inform a lot of things. It can inform public health. It can inform understanding just what's going on with this virus. And I would say if we just look, just look for a moment at just, what's going on in this country really specifically, I gotta say that I have no idea what we're doing, right? There's no question that when it comes to outbreak response, the United States has by far the most disappointing response of any countries. There are countries, even though that sounds, ridiculous, there are countries that actually done worse than we have, but it's very few, right? It's really, really remarkable how exceptionally poor we've been at responding to this. And I just wanted to have this slide here because it's really one for the history books back in April, right? The day where deaths topped 10,000 in New York City at the same time as pulling out of the World Health Organization. Not exactly what you want to do during a raging pandemic. Luckily, the stock market was up pretty well that day, so I guess it wasn't all bad news. But one of the reasons why we're doing really poorly with this is that I think we tend to think of these when, as hurricanes. There was this idea of when you just flatten the curve, it will all disappear. Of course it won't, right? That's what you see with hurricanes. The wind goes up, the wind goes down, and then it's gone. You need to prepare for new hurricanes, but that hurricane is not going to come back. The problem is with viruses don't do that, right? Viruses don't just magically disappear. That will just see for who can I infect and who can I not infect. So while you might see the curve going up, what's really important is that the virus doesn't know where it is on that curve. So the fact that it's going now is unbeknownst to the virus, right? So as soon as we just go back and pretend it's not there, of course it'll be back and it'll remind us very, very quickly, right? So viruses don't magically disappear, which we all know. I mean, it's ludicrous to even think that, right? But again, we can look at what's actually going on in this country. So some of the insights that we get, I'm just getting a couple of vignettes here and then I'm going to stop my talk. But the sort of things that we see when we're looking at the complex dynamic of the epidemic here in the United States is that it's not just one big epidemic that we have going on here, right? They're very distinct in different areas. And I'm just comparing some of our data from San Diego, where you can see there's a lot of different introductions coming in from all kinds of different places with not that much, ongoing local transmission early on in the epidemic, I should say. Compare that to New Orleans, for example, where you have a couple of really tight clusters and then super spreading events essentially leading to that entire outbreak that you have there. So very distinct profile of those two particular epidemics. If we just look at the New Orleans cluster specifically, we can show that this is actually all coming. When that got started, was all from viruses that were already circulating in this country. It didn't come from Europe, it didn't come from China, right? It was stuff that was already present there, which makes sense when we look at just travel too, is that most of this travel is actually coming from passengers within the United States. And I mentioned this, that we think super spreading event really early on. And as you know, back then, we had the Mardi Gras, for example, which probably worked as a super spreading event really early on in New Orleans. If we look at San Diego, it's a different picture. Again, not so many super spreading event, but constant migration of viral populations. And what's really interesting here is that if we're looking at the kind of viruses that we see circulating in San Diego and compare them to other counties in California, as well as Baja California down south, what we can see is that it's very connected to what's going on in Baja California, but much less so in, for example, Orange County and Los Angeles. So what that means is that if we are considering a place like Baja California and Imperial County, all of which are neighbors here in San Diego, is that it is essentially one big epidemic going on here. It's not just one and the other, it's really all connected. Importantly, we don't know anything about directionality. So we don't know if it's coming from Mexico to San Diego, from San Diego to Mexico. Assumption is that it goes both ways, right? It's not just one or the other. With that, I just wanted to thank a lot of collaborators. This is actually just a snapshot of these. All this work is extremely collaborative, both with local health authorities as well as academic institutions. And then all the heroes of my lab here who've really been doing an amazing amount of work during this whole pandemic, working in the lab, spending really, really long hours getting the sequence data. And also recent funding from the CDC as well as NIAID. And with that, I am going to stop the share here and happy to take a few questions. I can't hear Shruti again. Am I the only one who can't hear Shruti?
Please unmute your phone.
Oh, I apologize. Thank you. Thank you. That was a wonderful talk, Dr. Andersen. It was really, truly fascinating. I'm going to triage. There are a couple questions in our chat box, and one of them is, what do you think are important genomic and epidemiologic questions that remain unanswered?
Yeah, I think, so right now, just understanding how everything is connected and what actually drives that connectedness is important to understand, right? We believe that it's probably travel, but we don't really know. The genomics will allow us to see that. So understanding, again, emergence events as things flare up and understanding what are the drivers behind that is something we can do as part of these genomic analyses. And we're currently doing those. Other things is that as we are starting to come down with selection pressures from vaccines, very specifically, is that we would expect the viral diversity of the viral population to actually change in response to that, or potentially change in response to that. And for understanding that is really important, right? Because we're all worried about vaccine escape and what are the likelihoods of vaccine escape, and we don't know. There's been a lot of speculation around this based on the low evolutionary rate of the virus, but we really can't do that because until we have that vaccine, until we put that selection pressure, we don't know. But importantly, we then have to compare to what was before the vaccine. So the data set here describes that. To your original question of what about reinfections, right? Because reinfections is something that we're definitely going to start to see here during the fall and the winter in this country. But the question is it just lingering RNA? Is it the virus that actually reinfected? What's going on here, right? We don't know. And that is what genomics come in and can be really helpful because it helps us tell, is it the same virus? Is it different viruses? Different viruses, well, there must be reinfections, right? So it's going to be really, really powerful for that. And we are indeed, we're doing with San Diego County, for example, to investigate exactly those sorts of questions.
Outstanding. Thank you. We have a couple other questions. One of them, I think, was in relationship to just a few minutes after you began speaking. So I think it is in relationship to this talk, but just to confirm, it says reports in the popular press of cases and or viruses in sewage in Spain and in Italy. dating back to the spring of 2019, presumably should they not be believed?
Yeah, I don't believe it. Those are contaminants. There's no, and the same applies to like, there's been all these speculation around like a lot of circulation in this country dating back to December. I don't believe it. It's just the genetic data is really tight on like all of this getting started in, again, December, mid-November, early December of last year. Even if that pre-circulation hypothesis that I do favor, I would say I'm 55% on that one versus 45% on the other one, is that even if that one is true, it's got to be limited. Otherwise, it would have been detected. So things like that push this like way back in time, it doesn't make any sense, right? And it doesn't make any sense epidemiologically either, because we know how really easily this virus spread. We know the population exposures are super low. So I don't believe those stories.
Yeah. One more question I really think is a great question from Dr. Buckmeyer. Has SARS-1 disappeared completely, or are there pockets of the virus still in existence somewhere?
Good question. We don't know.
Yeah.
I mean, SARS was eradicated from the human population, right? But I can guarantee you that there is a bat out there, or a bat population or whatever, that have probably not exactly SARS, right? Because there's continuous evolution, but something that's basically SARS. Yeah, I'm sure it still exists out there.
Yeah, yeah. And Dr. Sandmeyer asks, is there enough diversity that virus sequence is useful for contact tracing on a university campus and identification of super spreader events?
Yes. So, and this is actually, so I didn't mention that because we're involved in many things, but we're actually doing exactly that. So as you know, there's been, university outbreaks, many places in this country. And we have one where we have been investigating with public health authorities, an outbreak that's now over 1,000 cases. And the belief was that, well, it's probably the virus came in multiple times and then caused an outbreak really rapidly, right? We're talking within just a few weeks, and then university had to shut down. We actually went in with genomics, and we have about 60 or 70 sequences from that outbreak by now. And what's really interesting is there's two main findings that we find. First of all, that introduction was not from the local circulation. So it's not from the local community. It's something outside the city of which we investigated this. The other thing we can show is that it's a single event. Like all the data so far just points to a single virus lineage that came onto campus, presumably via a single individual that was infected outside, right? Got onto campus, and then as part of the massive super spreading event is what caused what is now, counting more than 1,000 cases over just a few weeks, right? So that was definitely surprising to the public health authorities because that's not what they expected. But the genomic data clearly shows that that was what was going on. I suspect that what we are going to see from the White House super spreading event is probably similar, right? And that's things we are hoping to be involved in as well, is to look at exactly that. I don't think there was a lot of infected people at those events. I think it's just a single person, right? Single person, and given the right context, and woof, this just takes off.
Yeah, that sounds classical. Another question, good one as well. The SARS-CoV-2 seems to be optimally evolved. The infected is contagious before showing symptoms. How do you envision it to evolve, to coexist with humans? Yeah,
I mean, that's a really interesting question that I don't quite have the answer to, because again, the SARS, or traditional original SARS, wasn't very well adapted to human, but it actually did really early on in the epidemic there, did pick up some mutations pretty quickly, and then it transmitted bound better to the ACE2. If you look at SARS-2, it always seemed to actually it can already do that. Of course, it can always be better, right? But it's always already really good. So I don't think there's any strong selection pressures to push it in either direction, because there's been the speculation that it will evolve to essentially inactivate itself and it's going to be no big deal. It's going to be a common cold coronavirus. That's not going to happen based on just viral evolution, right? Because there's nothing pushing it in that direction from a selection perspective. On the other side, if you look at like, would it push it towards, worse clinical disease, increased transmissibility, for example, say there in the transmissibility question, there are some, yes, there is a selection pressure, but it's really small. And we have observed with the D614G, for example, probably does transmit slightly better, right? And slightly, we're talking maybe 10, 20% or so, which is difference that you can measure, right? But in the real world perspective, it doesn't really make a difference. But I don't really expect it to move a lot from there, right? Because the fundamental parts of the virus, like if it all of a sudden spreads really easily, like Ebola, for example, was hypothesized it would spread, airborne Ebola, right? It's not going to happen because making things like that happen takes a lot of mutations, very few of which are under selecting pressure, right? So because of that, I don't really expect SARS-CoV-2 to evolve in any particular direction to become more or less pathogenic, more or less transmissible of what we already have. It'll just continue to bounce around as it is. And what's also really important to remember here is that the viral population is huge, right? So even if you had one lineage that evolved some sort of trait that was either detrimental, certainly if it was detrimental, but even if it was under positive selection, is that for that to take over the larger size of the viral population is highly unlikely to happen, right? So when it comes to like, what do these mutations do? Given the neutral expectation we have now, I don't really expect much. That changes once we have the vaccine, or once if we have wide scale like antivirals that will be given to asymptomatics and people that might not take the full course, things like that. Once we come in with selection pressures, we might well see escape of these, vaccines and drugs, right? It's certainly a possibility because now the selection becomes really, really strong.
Fascinating. And I had one more question from me and the work that we do every day in healthcare epi and in the hospitals, the use of genomics to understand events of spread when the virus comes into our system or spreads within workers or between patients. Do you have any, of course, this is a national conversation in our field and many people who are interested in this work. And I wonder what kind of intel you may have, knowing what you know about the utility of this type of work and what time scale that should take place given pressures and mutation selections.
Yeah, I mean, it's not something I would expect to happen quickly, right? But the vaccine uptake is also not going to happen quickly, right? So I expect this is going to be a gradual change, and I would say that it requires pretty low population sizes, right? Given the tremendous amount of just infections we have going on right now, you need to get that lower before you're starting to see, these mutations actually playing a role given the selection pressures. So it's something that's going to take a long time. I would say in patients that are undergoing like remdesivir treatment or monoclonal cocktails for sure, is that it's going to be important to do serial sampling and sequencing, right? Because things like monoclonals, certainly if it's a single monoclonal, not actually a cocktail, you might expect just resistance to and escape, right, to evolve in the viral population within that individual. So from a basic perspective, it's important to understand those mechanisms. Now, I should say that people that undergo things like monoclonal cocktails, unless you're the president, right, who flaunts all safety measures you need to take here, unless you're him, if you get remdesivir or you get monoclonals, well, you stay in the hospital, right? Because you're really sick and you're getting these IV and you're not really at risk of spreading it to other people. So while resistance might develop in you, the likelihood of you transmitting it to other people is really, really low. And that's the only thing that matters when we're talking about transmission. If they're dead ends, right, they never get to go any further. Again, the situations of the president, I think, highlights it well that yes, while that might be true in principle and in theory, right, in practice, other things can happen. And doing things like taking his course and getting back home and taking off the face mask and driving home with people and exposing those, right, that is the kinds of events that can definitely lead pretty rapidly to escape mechanisms. But I assume they're going to be rare. Certainly hope they're going to be rare.
Right. Thank you so much. And I really pleasure to have you here, Dr. Andersen. And I don't see any more questions in our chat box. I'm going to turn it over to our host here, Sanghyuk Shin.
Thank you, Dr. Goldhill. So that was a fascinating way to end the symposium. And so on behalf of Dr. Semler and the rest of the planning committee, I want to thank everyone for joining us for this informative and timely symposium. I know you're really dying to go on to your next Zoom meeting. So Dr. Semler already acknowledged everyone in the beginning involved in putting this event together. So I won't go through the list again, but just really want to express my gratitude to the support team led by Lisa Sanchez of University Advancement. And of course, special thanks to our speakers who graciously took their time out to speak to us on the latest developments in COVID-19 dynamics. And thank you to our moderators for facilitating just the stimulating discussions all morning. And I think it's just striking how much our knowledge of this novel virus has grown and evolved during the past 9 months across multiple levels of disease dynamics, from host pathogenesis and immunological levels to the levels of viral evolution in human populations. But of course, there's clearly much more to learn. And so I just want to encourage everybody to please keep an eye out for future events and opportunities to continue this discussion coming out from the UCI Center for Virus Research and also the UCI Infectious Disease Science Initiative. And just speaking of upcoming events, I do want to briefly plug the 18th UC Irvine Immunology Fair that's coming up on December 3rd and December 4th. And this is organized by the UCI Institute for Immunology. So please keep an eye out for registration details. So with that, thank you everybody, and we look forward to connecting with you again in the near future.